Dear students, am I audible to you? Good evening, am I audible to you? Just a minute. Hello Akanksha, this is a good question. This is for everybody who's preparing for any kind of exams which inculcates your presence of biochemistry. So whoever wants to answer biochemistry questions in any type of exams, you should be able to answer this. Good evening Harish Rahul, good evening Himalani, good evening Akanksha Jain, good evening superoxide dismutase, Jwala and Life. So shall we begin? Am I audible to you? Are you able to see me? I'm not able to see myself, that's why I'm confused. Okay, so I hope you're able to see me. Okay, so let's start. Yes, this is the ultimate session that you would like to have because we have done this once and it was a huge hit. Many people had actually benefited from this particular session. I can assure you that this session will be maximally beneficial to you because in this session you'll be having 100 MCQs. As I name it, I give you my word, you will have 100 MCQs in them. No cheating in that, 100 MCQs, and you'll be having at least 500 facts to take home at the end of the session, 100 new concepts that you would actually learn in this particular session. You will have images in this session and you'll be learning about previous year questions also in this session. So this is Dr. Meenakshi Sundaram here. I'm an NEET PG educator. I'm an MBBS, MD and gold medalist MD exams, and I'm an educator for both biochemistry and microbiology. For those people who would like to actually come for a microbiology session also, I'm planning to conduct another session in the first or second week of May. We'll have one more session that is meant for biochemistry, meant for microbiology. And I have placed students every single year without any kind of difficulty in the top institutions every passing year without a fail. So I hope my students, if you're here listening to my classes, you also would actually go into the top institutions. And before we join, let me tell you about the free classes, free test calendar between April 25 and 30. The sessions that are available would be April 28th 10 p.m., 29th 10 p.m., 30th 7 p.m. and 30th 10 p.m. These are the different kinds of tests available for all the people who are preparing for the exams. And there is an All India mock test on May 1st, 2022 at 9 a.m. and that will benchmark your performance with All India rankers. And you have an Academy Gold, NEET PG subscription, and there's a launching of MBBS Professional 1. There is an Academy Lite for NEET PG. In all these areas, you can use code DRASM or DRASM Live to have a subscription with 10% discount and exclusive access to all my classes. And there's a plus subscription, iconic subscription, and in plus people you will be able to access NEET PG previous year question bank, and all the plus students after coming for one class you'll be able to answer the daily practice papers. These are the special class features, and this is the FMG high yield revision MCQ batch I'll be having in the month of May. I'll be doing this for the students for microbiology. So let's begin with the first question. Yes, Shivani Barot, good evening. So let's begin with the first question. Tell me, identify the methionine. Identify the methionine sparing amino acid. This is the first question of the day. Let's see how many of you can get the answer. Identify the methionine sparing amino acid. What is your answer? Ibiza says A. Anybody else? Shreya Mahapatra says A. Anybody else? Come on people, try giving answers. You will be able to make a huge difference for yourselves if you could answer. And remember, commit all your mistakes in this classroom. I repeat, commit all your mistakes in this classroom so that you will never have mistakes to do it in the exams. Okay, so Ibiza says A, Shreya says A, Navid Manzoor says A, Supraoxide dismutase says A, Shivani Bharat says A, Vishnuvardhan says A, Santosh Mahapatra says A, Akanksha Jain says A, Saurabh Banusali says A, Santosh Mahapatra says A, and the only person who has gotten the right answer would be Jwala. Remember, the right answer would be cysteine. What is cysteine? I repeat once again, listen very carefully. Cysteine is the amino acid who contains a free sulfhydryl group. Cysteine is the amino acid who contains free sulfhydryl group. Let me use a new slide here. If I have cysteine as a molecule which has a free sulfhydryl group, if I bring in another cysteine molecule that also has a free sulfhydryl group, both of them will come together to form a cysteine with a disulfide linkage, and two molecules of cysteine together will be referred to as cysteine. Cysteine is only an oxidized status of cysteine. Cysteine is only an oxidized status of cysteine. Cysteine's oxidized status is cysteine. Tell me, methionine is an essential amino acid or non-essential amino acid? Tell me if methionine is an essential amino acid or a non-essential amino acid. What kind of see, with one single question I made almost everybody make a mistake. Almost everybody made a mistake. So this is where you learn microbiology and biochemistry in such a fun fashion that you never make mistakes with simple areas. Even if they are very tough questions, you should be able to answer based on what you're learning. Tell me whether methionine is essential or non-essential. Excellent. Methionine is an essential amino acid. So essential amino acid means what? Will you be producing them in your body or you have to get them from the diet? Essential amino acids, will you produce them in the body or you get them from the diet? You get them from the diet. So that would mean to say that methionine has to be obtained only from the diet. Why? Because methionine is not produced by your body. But why are you requiring methionine? Because methionine is also a sulfur containing amino acid. Methionine is also a sulfur containing amino acid. But when you offer cysteine more and more, cysteine is also a sulfur containing amino acid. When you offer cysteine, the requirement of methionine will come down. This is called as methionine sparing effect of the amino acid called as cysteine. Let me put it again. If cysteine is available in abundance, cysteine, if it is available in abundance, then the requirement of methionine comes down. Requirement of methionine comes down. This is called as methionine sparing effect of cysteine. Not cysteine, it is cysteine. And also remember, cysteine is not actually active. It is an inactive molecule. It means the cysteine is actually being broken. It is being converted into cysteine. Tell me, cysteine as such, is it effective functionally, yes or no? I have told you that. Tell me, cysteine is active in which state? Is it active in the reduced state or the active state? Oxidized state? Tell me, cysteine is active in the reduced state or the oxidized state? Anybody? Excellent. Remember, cysteine is active in the reduced state. What is the oxidized state of cysteine? It is cysteine. Cysteine is oxidized and not mechanically or biochemically active. So it is biochemically inert. Now can anybody tell me the pharmacological utility of cysteine, which is reduced version? Can anybody tell me the pharmacological version of cysteine which is effective and useful? Pharmacologically, when will you come across cysteine as a useful compound? Not antioxidant, write this down. This is N-acetyl cysteine, which is used as an antidote. Excellent, Samir Parashari. It is used as an antidote for paracetamol poisoning. In paracetamol poisoning, your body would have actually utilized all the glutathione reserves. Glutathione reserves are exhausted. When you want to get back the glutathione again, now let me explain with one other picture. Watch very carefully. If you have gamma glutamyl cysteine glycine, this compound is called as glutathione. The chemical formula or the scientific name for glutathione is gamma glutamyl cysteine glycine. What kind of peptide is this? Is this a true peptide? Is this a true peptide? Is this a true peptide? This is called as a pseudopeptide. Why? Because all peptides will be made with alpha amino acid bonds. But this is made with a gamma bond. This is gamma glutamyl cysteine glycine. In that, SH group is present. So I will write the whole compound like this. I will zero in on this compound like this. I mark the whole circle as G with an SH. This is called as glutathione. Now, when I bring two molecules of glutathione together, they are applied on a compound called as X. X becomes XH2 reduced and removed from your body. While two molecules of glutathione will lose this hydrogen, they will form GSSG. Like how I told you, cysteine is not active in its oxidized state. GSSG is also not useful in its oxidized state. So when you want the glutathione to be returning back into the equation, if you have GSSG and if I add cysteine molecules, two of them, then two SH molecules will be offered so that you will be able to regenerate GSH. Why? Because GSSG is important for fighting paracetamol and to metabolize them. So remember this important logic. Cysteine is very effective in its reduced form. At the same time, when cysteine is available, methionine requirement comes down. Next question. Look at this question here. Look at this question here. A 40-year-old male is diagnosed with SLE and he presents with impaired compression or deformation abilities of a cartilage. So this one, she complains, this is a female and it's a her. A 40-year-old female diagnosed with SLE presents with impaired compression or deformation abilities of a cartilage. She complains of joint pain and swelling. Among the following extracellular matrix components, which is best suited in the given situation along with the impact it has on her cartilage? What is your answer? These are concept-based questions. You should be able to crack it if at all you are going to make it big in your exams. So do not miss out on these questions. Come on, what is the answer? Okay, Ibiza, 317, Zul, Saurabh, Banashali, you're all saying the answer is A and B. Anything else? Anything else? Okay, almost everybody says the answer is A. So the logic is that the patient is presenting with SLE and you're looking at the extracellular matrix components. There is a deformity in her cartilages and you're looking for the joint pain and swelling. You're asking for what can be right. The answer is obviously A. The reason is reduced proteoglycan leads to impaired resilience of the SLE cartilage. So proteoglycans are important for resilience. I repeat, proteoglycans are important for resilience. Proteoglycans are important for resilience. And one of the important reasons why they are capable of being resilient is they can accept water and they will be remaining in the form of a jelly. When they are capable of accepting them water and they are capable of remaining as jelly, then they are able to actually accept more amount of pressure. Now, when you look at the remaining options, elastin of SLE is unable to stretch. Excess collagen SLE causes joint stiffening. Laminin medial adhesion is interrupted, making SLE cartilage more diffuse. all of them, none of them are supposed to actually have resilience. Elastin, collagen, laminin do not contribute majorly to the resilience of the cartilage. Resilience is a result of the composition of the proteoglycans. And excellent people, all of you, those who got it right, you are amazing. You got the first question right. So the previous question, most of you made it wrong. Now all of you, or almost all of you, got it right. I repeat once again, this most important word in this question is the word called as resilience. Ability to take up more amount of pressure. So in terms of resilience, it is always related to the proteoglycan because it is capable of accepting water and being hygroscopic. Now let's go to the next question. Question number 3. We're going for question number 3. Drugs that block a virus from binding to which of the following type of molecules may turn out to be effective in preventing rhinovirus persistence? ICAM1, Syndecan, P-cadherin, L-selectin. This is like a integrated question. This is a combination of microbiology with biochemistry in terms of molecular biology and diagnostics with research ability. In 5 different areas, this question can be taken into consideration. Tell me, drugs that block a virus from binding to which of the following types of molecules can turn out to be effective in preventing rhinovirus persistence. For example, if a rhinovirus comes to sit in your nasal mucosa, it is not supposed to be persistent. For that, you are doing a drug. That drug will block which of the following molecules? ICAM1, Syndecan, P-cadherin, E-selectin. Which one is your answer? Excellent, Chakravarti. Chakravarti has gotten it right. Yes, Saif Alam, Zul. I have not made the PDF yet. If at all many students stay back and if they like to actually have the PDF at the end of the session, I can send the PPT file on my Telegram channel. You can search for my Telegram channel using Dr. ASM's DRASM's Forum for Infections and Biochemistry, Discussion for Biochemistry. You can search for it. I can put them on a PPT. I haven't made a PDF file. You can take this as a PPT file. If most students are available and most students are at the end of the session want the same question paper, I'll give you, okay? So the answer is ICAM1. What is ICAM1? It is intercellular adhesion molecule. Cellular adhesion molecule. Now here, let me make it very simple for you. When you focus on any kind of rhinoviral infection, a rhinoviral protein, capsid antigen, comes and binds to the ICAM1. This ICAM1 is important for keeping the nasal mucosa intact. Imagine the nasal mucosa to be like a mat. On the nasal mucosa, ICAM1 is present. If the rhinovirus capsid antigen binds to ICAM1, because of which the virus starts multiplying in the ICAM1, because of which ICAM1 becomes weaker and the nasal mucosa becomes weaker. When the nasal mucosa becomes weaker, your nose tries to fight against the infection by going for vasodilatation. And when that vasodilatation happens, the turbinates will increase in size. When the turbinates increase in size, then you call it as nasal blockade. At the same time, when the blood vessels are dilating, the space between endothelium can actually slightly increase, because of which liquid can come out. And that is the reason for rhinorrhea. So rhinorrhea and nasal blockade is because of your blood vessels response towards the ICAM1 binding. So if I use a drug which can prevent the capsid from binding to ICAM1, then you will not be suffering from all these kind of clinical features. That's all. Now let's go for the next question. Except ICAM1, no other adhesion molecules and endothelial cells of the host respiratory tract are welcoming of the rhinovirus. So the point is, ICAM1 is the only kind of surface molecule which can accept the rhinovirus. Next question. Now think carefully before you answer. Now this is question number 4, I believe. Yes, this is question number 4. If you would stay with me till 100 MCQs, I'll guarantee you that you will receive at least 100 points, 100 MCQs points, and at least 500 concepts. A hormone receptor placed strategically on the plasma membrane is best described as what? A hormone receptor placed strategically on the plasma membrane is best described as what? Is it peripheral protein? Is it lipid anchored protein? Is it integral membrane protein? Is it lipid raft? Is it glycolipid? Which one? Come on, people. Okay, many of you have given the right answer. The answer is integral membrane protein. A hormone receptor placed strategically on the plasma membrane. Strategically means what? If this is the membrane, you have a hormone receptor. So, if at all a hormone is the ligand has to come and bind to the protein present on the surface of the membrane. That cannot be a peripheral protein. It is supposed to be a protein that can travel through the membrane. So, it's an integral membrane protein. It cannot be a peripheral protein. It is not lipid anchored protein. Lipid raft is capable of moving from one place to other, and it is not a glycolipid. Look at this part here. The receptors in questions are transmembrane proteins. Transmembrane means they are able to move across the membrane once at least, and they are in communication with the external stimuli and the internal milieu. What is the external stimulus? The ligand or the hormone itself. When the hormone comes and binds, it will be able to make a contact with the ligand with the internal milieu. Since receptors are proteins, they are not glycolipids and they are not lipid rafts. Next question. Question number 5. Insulin causes decrease in the activity of what? Is it phosphofructokinase 1? Is it glucokinase? Is it pyruvate carboxylase? Is it acetyl CoA carboxylase? Which one? Excellent. Zul, Ibiza, Jwala, Himalini, Binay Yadav, all are saying C. Can you tell me why C? Remember one thing, insulin is an anabolic hormone. Anabolic hormone means it always tries to build something. Now there is an area where some confusion can happen. Listen very carefully. Except in case of glucose metabolism, except in case of glycolysis and gluconeogenesis, you will be having no trouble in understanding the concepts of anabolism and catabolism. Catabolism means what? You're trying to break a huge compound into a smaller molecule. Anabolism is using smaller molecules to build something very huge. Simple logic. Now, when you look for glycolysis and gluconeogenesis, these two are fuels. So, building a fuel called as gluconeogenesis is not anabolism, and utilizing the glucose in the form of glycolysis is not catabolism. So now, look at this part here. Let me tell you this. In concepts of metabolism, glycolysis is even though breakdown of glucose, it is only catabolism for the fuel called as glucose, but not catabolism in the bigger picture. So insulin supports glycolysis. At the same time, insulin discourages gluconeogenesis. So remember, anything that can increase the concentration of glucose in your blood will not be supported by insulin. So insulin always tries to bring down the glucose in your circulation. Logic number 1. Now in this picture, when you speak about phosphofructokinase, this is the rate limiting enzyme. Phosphofructokinase is the rate limiting enzyme of glycolysis. When glycolysis is supported by insulin, definitely this is increasing the activity of phosphofructokinase. Glucokinase is not actually having much of effect from insulin because hexokinase is mostly supported by insulin. Even if you take it into consideration, glucokinase is also a property of glycolysis. Again, insulin does not decrease the function. Acetyl CoA carboxylase. Can you tell me what is the function of acetyl CoA carboxylase? Can anybody tell me the function of acetyl CoA carboxylase? Yes. Okay, so okay, nice. So what else can you think of? No. Remember, acetyl CoA carboxylase supports beta oxidation is wrong. Acetyl CoA carboxylase is the rate limiting enzyme of Acetyl Coa Carboxylase is the first step towards fatty acid synthesis. Fatty acid synthesis would mean to say that you're trying to build a fatty acid using small molecules of acetyl CoA. You have smaller molecules of acetyl CoA and you take huge numbers of small molecules of acetyl CoA and convert them into a long chain fatty acid called as a 16 carbon fatty acid, which is palmitic acid. This is what happens. So this is a type of anabolism. I am building the fatty acid, it is anabolism. So it means to say that insulin will support acetyl CoA carboxylase also. And acetyl CoA carboxylase is also called as methionine CoA synthase. Methionine CoA synthase. Can anybody know? Please remember, this is not beta oxidation. This is not beta oxidation. This is fatty acid synthesis. Methionine CoA synthesis. Methionine CoA synthase, also called as acetyl CoA carboxylase, is also anabolism. Now, pyruvate carboxylase supports what? Pyruvate carboxylase is the first step towards gluconeogenesis. What happens in gluconeogenesis? We have pyruvate that gets converted into oxaloacetate. That oxaloacetate will become malate, and the malate again becomes oxaloacetate, which gets converted into phosphoenol pyruvate, and the remaining part of glycolysis will happen. So pyruvate carboxylase is important for gluconeogenesis. If it is gluconeogenesis, then it is not supported by insulin. So the only answer would be pyruvate carboxylase. Now let's go for question number 6. Look at this question here. A 2-year-old lean girl, the most important keywords are she is lean, she is 2 years old, she has increased frequency of micturition and is occasionally dehydrated. Now you're going for investigation. The urine is very dilute. If you look at the electrolytes, the sodium is at 155 milliequivalents liter, chloride is 125. Now you're performing gene studies. And the gene studies are showing the involvement of aquaporin 2 that has several transmembrane domains demarcated by beta turns. Pick the beta turn amino acids which are most likely to be mutated in this girl. Now this requires your understanding and your thought process. Tell me what can be the answer. Ibiza says E. Anybody else? Jwala says C. Okay. Anybody else? Yes. Sri is also saying C. Lazy Trip is also saying C. Very good. Anybody else? Saurabh says E. Okay. So there are some people who are saying E. Excellent. So Chakravarti says proline and glycine. The answer is glycine and proline. For that, you have to reiterate the question once again. They are asking you about the presence of aquaporin 2. Can anybody guess the diagnosis here? What is the diagnosis for this question? What is the diagnosis for this question? This is a case history. The answer is very simple. They are saying there is increased frequency of micturition, occasionally dehydrated. They are not talking about glucose at all. So the answer is excellent, Samir. It's a case of diabetes insipidus. In case of diabetes insipidus, you are attacking. Your concern is about aquaporin 2 channel protein. And that protein has several transmembrane domains. And their domains, look at this part. If I try to say several transmembrane domains, they go like this, like this, like this, like this, and like this. It means you are involving a super secondary structure. Super secondary structure means what? It is like a three-dimensional arrangement of secondary structures where you'll be able to have a three-dimensional space, and they will not be called as tertiary structures. They'll be unique because they are having their secondary structures coming together with each other. For example, we have alpha turn beta, we can have beta turn alpha, we can have beta loop beta, we can have beta barrel, we can have Greek key motif. Greek key motif. We can also have beta meander. All these things are examples for super secondary structures. Now when the loop has to loop out here, you are looking for a beta turn. The beta turn happens with how many amino acids? Can you tell me? Yes, it's a case of nephrogenic diabetes insipidus. Can you tell me the beta turn involves how many amino acids in the beta turn? When the beta is about to go for a turn, how many amino acids can be here? When you are looking for a beta turn, how many amino acids can I see? This is an important MCQ. Take it down. In a beta turn, during the turn area, you can expect 4 amino acids. Now imagine if this is an amino acid, 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 this is an amino acid. You can see the whole structure looks like a U. But if I want to get a V, what does it mean? I have to have a sharp angulation here. Sharp angulation if I want, do I need to have a long amino acid or a small amino acid? You tell me. Yes, the beta turns will have 4 amino acids. I'm telling you, if the amino acid chain is going to go for a turn like this area will be having a long amino acid or a small amino acid if I want a sharp projection. If I want a sharp turn, I need a small. Excellent. For that reason, glycine is important in a beta turn. At the same time, proline is not like a straightforward amino acid. It contains a ring structure, because of which the whole area can go for a sharper angulation. That is what is the logic. Have a look at this. Glycine and proline, a beta turn structure consists of 4 amino acids in which the first residue is hydrogen bonded to the 4th residue of the turn. If I have a structure like this is the first amino acid and this is the 4th amino acid, you will be able to have it 1, 2, 3, 4. So this kind of bonding may require for that to be present. Excellent. You can read the theory part here. Just have a look at this. This is for your benefit. Glycine residues are small, flexible. They are small, flexible, and they can give you a sharp projection, and they can assume a cis or flattened conformation. Transport proteins often have several membrane spanning domains. I told you transmembrane domains, right? They can go in and out of this particular membrane, and they will be having beta turns that allow them to exit and return back into the membrane. So what is the purpose of a beta turn? If at all you have a chain of amino acids, they have to again make a U-turn and they have to go back out into the they have to go back into the extracellular space. Again, they have to come back into the cytoplasm. This area, beta turn, is what we want, and the beta turns will be made up of glycine residues and proline residues. In nephrogenic diabetes insipidus, you have the kidney being less responsive to antidiuretic hormone, and that is excreted by the posterior pituitary. So treatment is difficult in part because these proteins are difficult to be controlled. So the paradoxical effect of some diuretics can be used. Tell me, what is the best antidiuretic hormone after antidiuretic hormone? Can anybody tell me who is the best antidiuretic after antidiuretic hormone? Look at this question here. Which is the best antidiuretic next to after ADH? No Chakravarti. That is a hormone you're saying. I'm asking you, what is the next best antidiuretic? Vasopressin. Vasopressin is the same as ADH. Vasopressin is the same as ADH. Take note of this. The answer is thiazide diuretic. Thiazide diuretic. Please make a note of this. It is a diuretic, but it has antidiuretic action at some level. That is what we refer to as treatment being difficult, and part utilizes the paradoxical effect of some diuretics in restoring sodium balance. I repeat once again, please take a note of this. No Ashik Raj, Saurabh Bansali, Lazy Trip. Did I say hormone? When did I say hormone? I asked you which is the best antidiuretic after antidiuretic hormone. The answer is thiazide diuretic. Did you hear the question, Prithvi Basu? I said which is the best antidiuretic. I didn't say which is the best hormone after antidiuretic hormone. I said which is the best antidiuretic after antidiuretic hormone. The answer is thiazide diuretic, and that thiazide diuretic is causing paradoxical antidiuretic action. You all can speak about aldosterone, that is a hormone. I'm asking you about a drug. So if at all you never knew this, please take a note of this. Thiazide diuretic can have, yes, thiazide diuretic can have antidiuretic effect. Next question. Now this is question number 7, I believe. 1 minute. Five. For every question, I'm giving you at least 5 concepts or 5 facts. So please be with me so that you will be able to use them in your exams. This is question number 6. Next we have question number 7. Tell me, positive nitrogen balance is seen in recovery from trauma, burns, pregnancy, stress. Is it A, B, B, C, A, C, B, D, 5, A, D? Which one? Thank you, Harish Rao. No problem, Prithvi Vasu. Prithvi Basu says A, C. Jwala also says 3. Ibiza says 1. Harish Rahul says 3. Himalini says 3. Madhu says 1. Any other answer? Sukha Karthik says 1. Samir says 3. Okay, the answer is 3. A, C. What is positive nitrogen balance? This is an indication for anabolism. Anabolism. What is balance, nitrogen balance? If I have a human being here, the amount of nitrogen going inside, say for example it is X, the amount of nitrogen going outside in the form of feces, sweat, urine, etc. If you are losing nitrogen, then that is called as output of nitrogen. Say this is Y. So if I say X minus Y, if the value is a positive answer, that is the amount of input is more than the amount of output, it means more amount of nitrogen is taken up by the body is building. If Y is greater than X, that is output is greater than the input, it means the body is losing more number of nitrogen. In people who are recovering from trauma or from any kind of viral infections, imagine if you have suffered from any kind of viral infection, after you have become better from the viral infection, if you are convalescing or you are getting better, during that period you will be having increased amount of appetite normally. Why? Because when you have a viral infection, you will start losing body weight because catabolism starts. Why? Because the viruses will be attacked by your macrophages and T4 and T8 lymphocytes. In that process, you will be having catabolism. I repeat once again, when macrophages are increasing their action, macrophages will be using their powerful weapon called as TNF alpha. TNF alpha is also referred to as cacectin. So when the macrophages are in action, the cacectin can act, because of which catabolism starts, body weight falls. So when you are recovering from trauma or viral infection, you will start vociferously eating things, because of which you will try to build your muscle mass. So you will be having a positive nitrogen balance. And pregnancy is a state where the lady has to build herself so that she will be able to build a small child also inside. So in these two conditions, anabolisms are important. So there will be a positive nitrogen balance. It means the amount of nitrogen going out is less than the amount of nitrogen coming in. Okay. Hormonal role. Insulin will be helpful for a positive nitrogen balance, while glucagon, epinephrine, norepinephrine, corticosteroids, everybody can cause a negative nitrogen balance. Next question. A middle-aged woman with poor mental health reveals that his older sister to have been admitted in the same institution as his. I repeat, a middle-aged woman, poor mental health, reveals that her older sister to have been admitted in the same institution as her. No similar history in their parents. The woman makes abnormal postures while sitting, and for some reason the consultant orders for a ferric chloride test in the woman's urine, and the test was positive. Name the amino acid to be found in her urine. Why not in burns? What is the question? In burns, you are actually losing your proteins faster and faster. And in case of burns, you are going for catabolism. Why? Because in burns, you have dehydration. So your body will be concerned about rehydrating itself rather than taking up amount of nitrogen. So, but when you're recovering from the same burns, you'll be having a positive nitrogen balance. Yes, what is the answer to this question? Remember, this is a case of phenylketonuria. It is not just a case of phenylalanemia. I repeat, you have hyperphenylalaninemia, which are of 5 types. Increase in concentration of phenylalanine can be of 5 different diseases, out of which type 1 is the most severe, which is phenylketonuria, where the original enzyme called as phenylalanine hydroxylase is deficient. In that area, phenylalanine can be deaminated to become phenylpyruvate. That can be again reduced to become phenylactate. I'll use this form here. Phenylalanine with the help of phenylalanine hydroxylase is supposed to become tyrosine. If this enzyme is deficient, or the coenzyme or cofactor called as tetrahydrobiopterin. If this is deficient, or the effect of tetrahydrobiopterin on the particular enzyme is actually not present, in any of these conditions, you will not be able to convert the phenylalanine to tyrosine. So phenylalanine can go for deamination to become what? It can become phenylpyruvate. This phenylpyruvate is a phenyl ketone, because pyruvate formula is CH3, C double bond O, COOH, a keto group is present. That can be reduced to become phenyl lactate. And the same phenyl pyruvate can actually become decarboxylated to become phenyl acetate. What is phenyl acetate? If you remove this carbon, you have only CH3COOH. All these are called as phenyl ketones. So this is a case of phenyl ketone urea. You may be having an abnormal smell, yes, but this is phenyl ketone urea where the ferric chloride test is positive. Have a look at this. Phenylalanine, is it essential or non-essential? Remember, excess amount of phenylalanine can be poor, can be dangerous for mentation. It can be dangerous for mentation and mental growth. So in case of phenylketonuria, when the patient has excess amount of phenylalanine accumulating, it can actually cause poor mental development. So low phenylalanine diets can allow the normal brain development, and you can have restricted food also. Can you tell me why artificial sweetener called as aspartame is contraindicated in phenylketonuria? Why the artificial sweetener called as aspartame is contraindicated in case of phenylketonuria? This was a very rare question, so remember this. The artificial sweetener called as aspartame is made up of phenyl aspartate. This can undergo deamination and it can ultimately come back to form phenylalanine, because of which it can add to the load of the patients. So this is contraindicated in patients who are suffering from that of hyperphenylalinemia. Okay, now let's go for question. Serine. Serine is produced in the human beings from what? Serine is produced in the human beings from what? Not B. Anybody else? Okay, not C. The right answer is serine can be easily formed from glycine. Do you know what is glycine? CH2 COOH on one side and NH2 on the other side. This is called as glycine. Now if I have something here, NH2 CH CH3 COOH, this is called as alanine. And if I have one more molecule like this, NH2 CH CH2OH, then I have COOH. Hydroxymethylglycine is called as serine. So glycine can give rise to serine, serine can give rise to glycine. This is what I would want you to remember for these exams. So this is one particular slide that is very helpful for you. What is the function of serine? Serine is very helpful for phospholipids in the form of phosphatidylserines. Glycine is important for glutathione formation along with cysteine. Glycine is also important for creatine formation. Methionine also adds to methionine cycle, where it will be important for methylation of DNA, RNA, proteins and lipids. You will be having glycine that is important for ATP, GTP synthesis in the form of nucleic acid. Why? Because glycine is directly incorporated into the purine area. I repeat, this is an important MCQ area. This topic will be a very rich source of MCQs in your exam. Yes, serine hydroxymethyl transferase is the enzyme through which glycine can be formed from serine, or serine can be formed from glycine. Now take this slide very seriously. I repeat once again, the whole molecule of glycine can be incorporated into the purine. The purine ring that we say, right? When you draw the purine ring, it will be like this. You'll be having two rings coming together. Now in this, the whole area will be offered by glycine. So glycine is very essential for purine synthesis. It means for the synthesis of ATP and GTP, you will be requiring purine. And the nucleic acids are not synthesized without purine. And the folate cycle is important for NADPH, NADH and ATP. Now methionine cycle is important for methylation of DNA, RNA, proteins and lipids. Okay, next question. true about carbamyl phosphate synthase. Is it a mitochondrial enzyme? Is it a cytosolic enzyme? It catalyzes condensation reaction. All are true. None of them are true. Which one is the right answer? Correct, Prithvi Basu. Glycine plus arginine plus that of methionine together will be called as creatine. Zul says A, Lazy Trip says D, Prithvi says D, Chakravarti says A, Ibiza says A, Himalini says D, Jwala says D, Saurabh Banushali says D, Yashwardhan says B, Ibiza says D. Okay, now watch very carefully. This synthase is supposed to be a true synthetase. What is the difference between synthase and synthetase? If anything is synthesized without ATP, it is called as synthase. If you require ATP in the formation, you call it as synthetase. Now this carbamyl phosphate synthetase is actually of two types. CPS1 is present inside the mitochondria, which is important property of urea cycle. If it is synthetase 2, that is present in the cytosolic activity, it is important for pyrimidine synthesis. So both A and B are true. So for pyrimidine synthesis, you will be needing cytosolic enzyme, and if it is for urea cycle, you need mitochondrial enzyme. And it is catalyzing condensation reaction. Yes, when you have a carbon dioxide plus ammonia, this ammonia plus carbon dioxide together will form carb-O-moil phosphate. So this carbon dioxide and ammonia are synthesized together with the help of ATP. So this comes under which class of enzymes? Anybody? Excellent, Prithviraj Basu. Excellent, Chakravarti. Very good. Madhu, very good. But no, not lyase. You came so close, but not lyase. Which class of enzyme? Which class of enzyme does CPS come under? Excellent. It is an example of ligase. So can you tell me how many classes of enzymes are there? How many classes of enzymes are there? Excellent. So we have 7 classes. OTHLIL tra. This is ethyl tra, oxidoreductase, where there is a transfer of either oxygen or hydrogen or electron only. Next is transferase, transfer of any group other than oxygen, hydrogen, electrons. For example, a methyl group, an amino group, or a carboxyl group rarely, or you can have a sulfate group or a phosphate group. Examples of transfer. H is hydrolase. Hydrolase, tell me, hydroxylase comes under which class among these? Hydroxylase comes under which class? Hydroxylase enzymes will come under which class? OTHLIL. This is 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7. Can anybody tell me hydroxylase comes under which class? Yes, Prithvi Basu, Bharat Krishna are all saying that it is a lyase. But the mistake is that you haven't understood the concept of hydroxylase. Saurabh Banushali is correct. Hydroxylase means, for example, I give you an example here. You have phenylalanine. This phenylalanine will be having a carbon, 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 and each carbon will be bound to hydrogen, 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 and hydrogen. When I am saying I am hydroxylating phenylalanine to tyrosine, do you know what I am doing? I am adding a singlet oxygen between this carbon and this hydrogen, so that this carbon will become a hydroxylated carbon. So what have I done? I have added only a single oxygenase. So hydroxylases are examples of monooxygenase. Addition of oxygen or removal of oxygen, all of them come under class 1 called as oxidoreductase. Hydroxylase is not going to break any bond, but hydrolase is going to break a bond with the help of water. Lyase is going to break the same bond without using water. I is isomerase. Can you give me an example of an isomerase where B12 can be effective? Always gets confused. Huh? So did you understand this? Hydroxylase means creating a hydroxy group in a molecule. Hydroxy group creation means you are introducing a single O between the C and H. So introducing a single O between C and H means it is a monooxygenation. Did you understand this, Prithvi Basu? Did you understand this, Prithvi Basu? Why hydroxylase is oxidorreductase? Are you in there? Do you want me to repeat again, or have you understood this, Prithvi Basu? Yes. Now, isomerase in a place where B12 can be involved. I repeat, you have you have the vitamin B12 in different forms. Vitamin B12 is cobalamin. The cobalamin has active forms in the form of cyanocobalamin, methylcobalamin, adenosylcobalamin, aquacobalamin having water, and hydroxycobalamin having OH group. So here you remember the injectable form is adenosyl, the oral form is cyanide, the storage form can be adenosyl, and the aqua hydroxycobalamins are variants. Now methylcobalamin is the most common way of storage inside the body. Now methylcobalamin is important for transmethylation reactions. Methylcobalamin is important for transmethylation reaction where a compound X becomes methylated X because methyl B12 became B12. Now this is the methylation area. But adenosyl B12 is important for one activity. Can you tell me what it is? Adenosyl B12 is important for the whole process where a propionyl CoA is ultimately trying to become succinyl CoA. This succinyl CoA is the pathway through which it can enter into TCA cycle. So propionyl CoA will first get a carbon dioxide to become methyl malonyl CoA. That methyl malonyl CoA will be initially D type. Then it becomes L type. Then that rearranges to become succinyl CoA. In that, the isomerases and mutases are examples of isomerase. The next L is ligase. And the last seventh one is translocase. Translocase is the translocase. No, there is no TCA cycle isomerase. Now the seventh class of enzyme is translocase. So the CPS will come under ligase, which is class 6 of enzymes. Can you give me an example of a translocase? Can you give me one example of a translocase? Let me ask you a question. Tell me, bongregate in relationship to translocase. What is the relationship? Okay, carnitine palmitoyl transferase 1 is what you're calling it as a translocase. And that carnitine palmitoyl transferase can have enzymatic activity. That is why it is capable of binding with the carnitine. It can transfer. So can anybody tell me this relationship here? Can anybody guess the relationship here? The example of translocase is ATP, ADP, translocase present in your electron transport chain. Imagine this. In your inner mitochondrial membrane, you will be able to have these kind of cristae. You have complex 1, you have complex 3, you have complex 4. Then you have the FOF1 ATP synthase complex, which is called as complex 5. Now, apart from that, there is an equivalent called as complex 6, which is called as ADP ATP translocase. If this is not functioning, ADP cannot come inside. ADP is needed because in the ATP synthase complex, ADP enters inside along with that of inorganic phosphate and comes out as ATP. The ATP synthesized the mitochondria will leave through the pathway to the cytoplasmic area. So ADP comes inside, that becomes a substrate for the ATP synthase complex. It goes out of your body it goes out of the mitochondria as ATP. This is an example of a translocase which can be inhibited by two important compounds. It can be one of them being atractyloside, which can inhibit the ATP ADP translocase, and the other one is bongregate bongregate or bongregaic acid, which is coming from the bacteria called as Pseudomonas. Both are capable of blocking this particular complex, because of which ADP is not coming inside, ATP is not forming. So ATP synthase complex comes to a halt. So the whole mitochondria comes to a halt. Mitochondria will come to a halt, because of which you will not be able to synthesize ATP. No synthesis of ATP. That is the beauty of ADP-ATP translocase enzyme. Okay. Now look at this question here. This requires your understanding. Take your time and give me an answer. What is the answer for this question? A middle-aged premenopausal female presents with menorrhagia, anemia, with poor response to iron supplementation. Which of the following results is most likely if the patient has a hemoglobinopathy? Think of some hemoglobinopathy, and that hemoglobinopathy is not responding to iron supplementation for the anemic status. So you understand there is a possibility of having an anemic status. Of the following, which one can be closer to the answer? Two proteins of normal quantity visualized by Western blotting, several proteins and two red proteins separated by native gel electrophoresis, two labeled bands a slight distance apart after SDS gel electrophoresis and reaction with labeled antibody to alpha and beta globin, several proteins but only one red protein derived by HPLC, a reddish mixture of proteins retained within the dialysis membrane. Prithvi Basu says C, Harish Rahul says D. Can you first guess what is the hemoglobinopathy we are discussing about? What is the hemoglobinopathy that we are discussing about? Which hemoglobinopathy am I discussing about? Any guess at all? See, these are some bouncers. It may be difficult for all the people to answer. So don't worry about it. Just be with me. I'll explain it to you so that you will not be able to find it difficult in your exams. That's all. Jwala says beta thalassemia. Harish Rahul says thalassemic trait. HBE, sideroblastic thalassemia trait. Saurabh Banusali, sideroblastic anemia is the word you use in case of poor synthesis of the heme, where the B6 may be deficient or ALA synthase may be deficient, because of which delta aminolevulinic acid is not synthesized. The first step of heme synthesis is blocked, because of which you may have, that is in B6 deficiency, you may have sideroblastic anemia. The answer is C. What you are performing is actually a polyacrylamide gel electrophoresis. Here you are looking at a mutation. In the mutation, this mutation is when one alpha or beta globin allele alters the primary amino acid sequence of the encoded globin peptide. See, if a gene is mutated, and in the gene if there is a mutation, then when I look for a primary amino acid activation where you have the peptide bond here, in that the primary amino acid sequence itself is altered. If the amino acid change alters the charge of the peptide, when the charge is altered, then the hemoglobin tetramer assembled with the mutant globin peptide has a different charge. So because of which electrophoretic migration will be different than that of the normal hemoglobin tetramer. So what you can have is two labeled bands a slight distance apart after SDL gel electrophoresis, and the reaction with the labeled antibody to alpha and beta globin can be noticed. How? Have a look at this. See, these are the questions we are doing so that we'll be able to go through them faster. Please have a look at this. In case of electrophoresis, you will be making a chamber. The chamber will be filled with a lot of units of molecules called as buffer solution, and you will be having a platform on which a particular solution can be placed. An electrical current will be applied on both sides depending upon the charges present on the molecule and the sizes present of the molecule. The movement of them will be able to create a pattern. This pattern is called as electrophoresis. Now look at this particular paragraph. The electrophoresis of native hemoglobin produces two species, two bands, each retaining its heme molecule in red color. if the hemoglobins were first denatured into their alpha and beta globins, as with SDS polyethylene electrophoresis, see SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. If you perform that, then that will be able to denature. This will denature the alpha and beta globin chains. Now you will be having similar size of alpha or beta globin peptides if the denaturation happens. And when the denaturation happens, if both of them are alpha and beta properly present, then they will be moving closely together as two colorless bands. So if they are moving together as colorless bands, then they are definitely not something wrong. So you would have gone for option A. Option A speaks about the presence of only two normal bands, alpha and beta. But now look at this. Because sodium dodecyl sulfate, a detergent, covers the protein surface and causes all the proteins to be negatively charged. What is the point here? See, you have, now let me make it as simple as possible. It may be very long and difficult. You have alpha chain, beta chain. You have two alpha chain and one beta 1 and beta 2 chains that are present in case of any kind of normal hemoglobin. Now they will have different charges depending upon which chain it is. Now, if at all you run them on electrophoresis, you'll be producing two different bands. Alpha will have its own band, beta will have its own bands, because both of them will be having different charges. Now, when you have thalassemia, because of any kind of mutation, mutation means gene is altered, because of which amino acid present on the surface of the protein is altered, because of which charges may be altered. But when I perform a sodium dodecyl activity, the sodium dodecyl sulfate will come and cover both of them, because of which any charge that is present on their surface will become neutralized. The charge will not be present, they will be masked. When the charge is masked, then the movement is purely because of what? I'm asking you a simple question. Electrophoresis chamber, electrophoresis generally happens either with the difference in the charges or difference in the size. Now when I use SDS and I cover all the protein negative charges, then what will be the only reason by which I can actually move? The movement will be based purely on what? The movement will be purely based on what? If I have covered all the charges on the hemoglobin molecule, then the movement of the molecules will be based on which property? Yes, you don't say weight, it is based on size. Please take note of this. So high performance liquid chromatography can be used to go for a separation based on charge. So this is what I want you to remember about this question. Okay, now look at this question. Which of the following amino acid residues of albumin is neutral under regular physiological conditions in blood? What is your answer? Niranji Moon, we have blocked the charges using SDS page. So the only way by which now you can differentiate them is based on the sizes. Okay, what is the answer for this question? Which of the following amino acid residues of albumin is neutral under regular physiological conditions in blood? Prithvi Basurai says Ye, Saurabh says histidine, Himalini also says histidine. So now this is the place where you haven't understood the question. The question looks extravagantly complicated. They are just asking you for a neutral amino acid. The simple question is they're looking for a neutral amino acid. They have twisted the statements in such a way saying that which are the following amino acid residues present on the surface of albumin. Imagine if this is the albumin. On the surface, we are looking at all the amino acids. Which amino acid is neutral under physiological conditions? So we are looking for a neutral amino acid. Then what is the answer? If I am looking for a neutral amino acid, what is the answer? No, please remember the classification of amino acids. Let me tell you, shall I tell you the classification of amino acids? Is it okay if I tell you the classification of amino acids? Based on the polarity? Is it okay if I tell you the classification of amino acids based on the polarity? No, again you're wrong. So listen carefully. Non-polar aliphatic amino acids are GAV-LIM, glycine, alanine, valine, leucine, isoleucine and methionine. These are the non-polar aliphatic amino acids. Who are the aromatic amino acids? It is phenylalanine, tyrosine and tryptophan. Because they are aromatic, they are also supposed to be non-polar. But tyrosine contains a hydroxy group. So among the three, the most polar aromatic amino acid is tyrosine because it has a hydroxy group. Point number 1. So I have told you about the non-polar aliphatic amino acids and I have told you about the non-polar aromatic amino acids. Now let me tell you about the polar amino acids. These polar amino acids can be either charged or they can be uncharged. In that we focus on charged amino acid. The charged amino acid will be aspartic acid, who contains more carboxyl group than the amino group. You have glutamic acid, which also contains more carboxyl group than the amino group. These are positively charged amino acid, polar. And we have negatively charged, these are negatively charged amino acids who are polar. Who are the positively charged ones? Histidine is a charged amino acid and arginine is also a charged amino acid. Lysine is also a charged amino acid. So HAL, histidine, arginine, lysine, they are charged and they are polar and they are basic amino acids. While aspartic acid, glutamic acid are also charged and they are acidic amino acids. While the uncharged one will be the neutral amino acid coming out of aspartic acid called as asparagine. Glutamic acid will go for neutralization to become glutamine. So glutamine and asparagine. What happened here? Look at this very carefully. In case of glutamate, you will be having a COOH here and will also be having another COOH here. There are two carboxyl groups present. Now, among these two carboxyl groups, if I am going to attach one more amino group, and if this amino group goes to the COOH and that becomes CONH2, then this is called as amide bond. The moment the amino group has blocked the carboxyl group and has converted it to an amide, then this carboxylic effect is not available. In glutamic acid, what happens? You have two COOH groups plus one amino group in case of glutamic acid. The same in case of aspartic acid also. But in glutamic acid and aspartic acid, if I attach an amine here, because of which I produce amide, then the glutamate becomes amide form called as glutamine. The effect of COOH has been nullified. The same in case of aspartate, which becomes asparagine. So please take it into consideration that glutamine and asparagine are the examples of neutral amino acids who will be remaining neutral under physiological condition. Histidine is a basic amino acid. Glutamate is an acidic amino acid. Aspartate is also an acidic amino acid. Arginine is a basic amino acid. Among the following, who has? Did you all understand what I'm saying? Hello students, did you understand what I'm saying? Yes. So now let me tell you one more thing. Histidine, because Prithvi Basu was having a lot of attraction towards histidine, just like other people also who chose histidine. Histidine, even though it is a basic amino acid, what are the other features of this? Histidine, like asparagine, both of them are considered as conditional essential amino acids. Conditional or semi-essential amino acids. Only when the demand is very high, as in case of childhood, where a child is trying to grow into adult, in that period they may be required in the diet. So you may require more, so you want them in the diet, you call them as conditional or semi-essential amino acids. Point number 1. Apart from that, what are the other things you have to know? What are the other things you have to know? Histidine has a pKa value. Histidine has a pKa value which is closer to the pH of the body, closer to physiological pH. What is the physiological pH? It is 7.35 to 7.45, and the histidine's pKa value somewhere at 6.8. So histidine becomes the most efficient buffer. It is the most efficient buffer. Why? Because anybody who has a pKa value which is closer to the pH of the physiological medium, then that can act as an efficient buffer. But who is the most effective buffer? Who is the most effective buffer? The meaning of the word efficient and effective are different. The most efficient buffer is histidine buffer, but the most effective buffer will be your bicarbonate buffer. Why? Because even though bicarbonate buffer system's pKa value is somewhere in the range of 6.1. See, this is 6.1, histidine is 6.8. 6.8 is closer to 6.8 is closer to 7.35 compared to the 6.1. Even though it is 6.1, very far away from 6.8, bicarbonate is the most abundant buffer. So the effect of the abundant buffer is always high. Ultimate result is higher. So the effective buffer is bicarbonate, while the efficient buffer is histidine. Is that clear? Okay, now let's go for this question. Now answer this. Yes. Which of the following amino acids is involved in the pathways leading to neurotransmitters and or melanin? This is an intelligent question. Try to answer this. Madhu says 5. It is C, G. Very good. What else? Padmaja says 2. What else? Saurabh says 5. Prithvi Basu says C, G. Excellent. So, you are speaking about neurotransmitters. Neurotransmitters are what? You divide them into excitatory neurotransmitters and inhibitory neurotransmitters. When you come across inhibitory neurotransmitters, you have glycine, GABA, taurine. For excitatory, you have N-methyl D-aspartate and you have glutamate. These are the ones which we focus on. And for melanin, it comes from that of tyrosine. So on that basis, G is definitely an answer. Now, remaining part. The glutamate, yes, it is present. Arginine, no. Cysteine, no. Leucine, no. Proline, no. Serine, no. What about the glutamate? The glutamate on decarboxylation can become gamma amino butyric acid, which is this one. Glutamate by itself can act as a neurotransmitter, which is this one. So the right answers would be glutamate and that of tyrosine. Okay. Let's go for the next question. During prolonged starvation, the initial breakdown of proteins happen in which form? Skeletal muscle, brain protein, hepatic storage proteins, cardiac muscles. Which one? Yes, Himalani. Why is there a different question here? Serine can be a source. Serine can be a source of serine. Serine can be a source of glycine. But look at the options here. In the given options, can you come back to anything else? I have chosen to choose C and G as the answer. If there is an option which contains C, F and G, you can go for it. But I have given you only glutamate and tyrosine. And if you want to choose serine, you have to go among all of them. But all of them is wrong. A, B, C, D is wrong. B, C, D is wrong. That is why the best answer would be C, G in the given question. C, G in the given question. Okay, so I can see during prolonged starvation, the initial breakdown of proteins happens from where? All of you got it wrong. It is not a hepatic storage protein because it will be the last group of proteins to be lost. The answer is skeletal muscle. Skeletal muscle contains maximum abundant amounts of alanine. Now comes a doubt. You said prolonged starvation. Yes, prolonged starvation. In prolonged starvation, gluconeogenesis will start. But gluconeogenesis initially will begin with the skeletal muscle proteins. The answer is A. Did you all get this? See, I am not asking about glycogenolysis or gluconeogenesis per se. I am asking you which protein will be broken down first to supply for the initial activity of glucose. Yes, so the answer is skeletal muscle, not anybody else. Among this, which of the following is a lyase? Anybody? Okay. So we have two cycles. One is Cori cycle, the other one is Cahill cycle. According to what Prithvi says, if in the liver you can produce glucose by gluconeogenesis and that goes to the muscle, in the muscle glucose is broken down into pyruvate and that pyruvate can become lactate and the pyruvate and lactate will be lost into the blood. Through the blood it can reach your liver and in the liver through gluconeogenesis from pyruvate and lactate again you can produce glucose. This is called as Cori cycle. Now again in the liver versus muscle, if the muscles are broken down because of extreme starvation, the alanine present in the muscle will undergo deamination to become pyruvate. Now that pyruvate will enter into the circulation. Through the circulation it can go to the liver. In the liver the pyruvate will undergo amination. The pyruvate will turn into alanine. That alanine again will be thrown into blood. Through the blood it reaches your muscle. This is called as Cahill cycle. Okay, what is the answer to this question? Which of the following is a lyase? Niranji Moon, we are speaking about breakdown of proteins only. We are speaking about breakdown of proteins only. What you are asking me is glycogenolysis. We are not at all discussing about gluconeogenesis or glycogenolysis. We are just saying if at all it is needed, if proteins are needed, the proteins will come from where? The answer is skeletal muscle. But if you have to satisfy your question, whenever you are having postprandial state, if you have crossed 3 hours after having food, from the 3 hours of after having food till the next 12 hours, you will generally be using glycogen breakdown for your supply. After 16 to 17 hours, gluconeogenesis will take over. So glycogenolysis will supply the glucose and then gluconeogenesis will supply the glucose. Here what is the answer? Which of the following is a lyase? Transaminases are transferring amino group. They are called as aminotransferases. So they are coming under class 2. How can they be lyase? Lyase means they have to break a bond. Here the breaking of a bond is actually followed by a formation of a new bond. Excellent. The answer will be aldolase B. Aldolase B or aldolase A. What does it do? See, aldolase B or A. Aldolase A is found in glycolysis. What happens here? In glycolysis, fructose 1,6 bisphosphate will be broken into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. In case of aldolase B, you will come across aldolase B in fructose metabolism. In fructose metabolism, you will be having only fructose 1-phosphate. Now this fructose phosphate will be broken down into glyceraldehyde plus dihydroxyacetone phosphate. But both of them are trying to break the bond, because of which a 6-carbon compound, 1, 2, 3, 4, 5, 6. I will break it here. One becomes aldehyde, the other becomes ketone. This is what is the property of the lyase. Who are the other lyases for your information? Other lyases are arginosuccinate lyase that you find in the urea cycle. ATP citrate lyase, you will be finding them in the cytoplasm, and you will be having fumarase in case of your breakdown of fumarate. HMG-CoA lyase in terms of ketone body synthesis. Ketone body synthesis. These are the different types of lyases you will come across. Next question. Enzymes that need biotin as coenzymes. Which one? Which are the enzymes who need biotin as the coenzyme? Okay, they're saying 2. 2 is what? A, C and E. Okay, anything else? Any other answer? Raman Thakur is saying 3. Niranji Moon is saying all carboxylation reaction. So Himalani, Padmaja, Ibiza, Niranji, Raman, Prithvi. Okay, except Raman, everybody has said 2. So Raman is the only person who has gotten the right answer. Remember, biotin acts as a coenzyme only in a few carboxylation reactions. There are some carboxylation reactions which are capable of happening spontaneously. And there is one more vitamin called as vitamin K that also helps in carboxylation reaction. And this vitamin K, remember carboxylation reactions. So vitamin K carboxylation reactions are also available. Now look at this. Acetyl Coa Carboxylase is the rate limiting enzyme of fatty acid synthesis that requires biotin. After that, pyruvate carboxylase, the first step towards gluconeogenesis, that will also require biotin. But when you look at pyruvate dehydrogenase, this is not a carboxylation reaction. I repeat, pyruvate dehydrogenase helps in decarboxylation. I repeat, pyruvate dehydrogenase converts pyruvate into acetyl CoA. This is an example of a decarboxylation, and this is not carboxylation. And proconvertin carboxylase, proconvertin is a coagulation factor. So any kind of coagulation factor will be always coming under vitamin K. Now let me tell you, there are something called as vitamin K dependent carboxylation reactions. Vitamin K dependent carboxylation reactions, where you will be expecting gamma carboxylation of glutamic acid residues. Shall I explain why this happens? If you want. What is the purpose of this gamma glutamic carboxylase? Do you want me to explain the gamma carboxylation act if you want? Okay, now look at this. This is glutamic acid. Now, under regular situations, you may expect this to have this particular H gone, then you will be having COO minus here. Now, what is gamma? See, carboxyl group has more power than the amino group. So, this will become alpha carbon, this becomes beta carbon, this becomes gamma carbon. Now, when I carboxylate the gamma area, you will be getting one more carbon dioxide. Now, when you have one more carbon dioxide, this is called as gamma carboxylated glutamic acid. Imagine if this is a protein. In that protein, this is the glutamic acid. The glutamic acid's gamma position has been carboxylated. Now there is a -1 charge here, -1 charge here. Between the -1 charge and -1 charge, a calcium with a 2 charge can come and bind. So this is called as calcium trapping ability. Calcium trapping ability. Previously what we had was glutamic acid residue that is present in the protein. And when the glutamic acid residue got the gamma carboxylation, that is referred to as GLA. So you call them as GLA proteins. GLA proteins are those who are getting gamma carboxylated with the help of vitamin K. On that basis, who are the proteins who get gamma carboxylated with the help of proteins? Gamma carboxylation of glutamic acid residues will cause activation of proteins like factors 2, 7, 9, 10. That is, among them you will be having proconvertin. Then you will be having proteins C, S, Z. Then matrix GLA protein. Then you will be having nephrocalcin and osteocalcin. All these reactions will require vitamin K for gamma carboxylation. Now, acetyl CoA carboxylase, pyruvate carboxylase, malenyl CoA activity are all concerned about biotin activity. Okay. So now let's go for the next question. Look at this. Acetyl Coa Carboxylase, Propenyl Coa Carboxylase, Pyruvate Carboxylase, Urea Carboxylase, Geranyl Coa Carboxylase, not important for you. Methylcrotonyl Coa Carboxylase, not necessary for you. These two are not important for you. These are important in botany. But acetyl Coa Carboxylase, Propenyl Coa Carboxylase, Pyruvate Carboxylase, Urea Carboxylase are all important in case of they are all requiring biotin. Next question. Try to answer this question. TPP required in which reaction? Thiamine pyrophosphate is the active form of vitamin B1. This is present in pyruvate dehydrogenase complex. This is a complex where the pyruvate dehydrogenase enzyme requires thiamine. Then we have alpha keto glutarate dehydrogenase complex. Then we have branched chain keto acid dehydrogenase complex. These are all dehydrogenase enzymes which can also perform a decarboxylation activity. Then thiamine pyrophosphate can be helpful for trans-ketolase enzyme in case of HMP shunt. They are also important for tryptophan pyrrolase enzyme, tryptophan pyrrolase enzyme, which is helpful for tryptophan catabolism. These are the 5 major areas where thiamine pyrophosphate is important. Now, if you want to know the knowledge in the reverse fashion, in pyruvate dehydrogenase complex, what are the cofactors present? In pyruvate dehydrogenase complex, you will be having 3 enzymes and 5 vitamins. The 3 enzymes are pyruvate dehydrogenase by itself. That is, in the pyruvate dehydrogenase complex, you will be having pyruvate dehydrogenase. Then you have dihydrolipoyl transacetylase. Then you have dihydrolipoyl dehydrogenase. 3 enzymes. Then we have 5 vitamins, which is B1 in the form of thiamine pyrophosphate. We have B2 in the form of flavin adenine dinucleotide H2 reduced. B3 in the form of nicotinamide adenine dinucleotide reduced H+. Then we have B5, that is pantothenic acid in the form of coenzyme A. Then we have lipoic acid. These are all the components present in pyruvate dehydrogenase complex, also found in alpha ketoglutarate dehydrogenase complex, also found in branched chain keto acid dehydrogenase complex. Is that clear? Have I answered all your questions, Saurabh Bhanushali? Okay, you're welcome. The enzyme deficient in maple syrup urine disease is which one? Yes, excellent. The answer is branched chain keto acid dehydrogenase. Now, can you tell me which vitamin deficiency can lead to maple syrup urine disease? Which vitamin deficiency can lead to maple syrup urine disease? The answer can be biotin. Biotin is important for carboxylation reactions. At the same time, thiamine pyrophosphate, flavin adenine dinucleotide, nicotinamide adenine dinucleotide, lipoic acid, pantothenic acid, they also can cause a similar activity of the maple syrup urine disease, but the most important vitamin always will be biotin. No, not thiamine. In case of branched chain keto acid dehydrogenase, you will be requiring all these components TPP, FAD, NAD, lipoic acid and pantothenic acid is coenzyme A. But the other vitamin that is required for that the vitamin that is deficient in maple syrup urine disease is biotin. Now look at this picture here. See, what are the branched chains? Leucine, isoleucine, valine. Leucine, isoleucine, valine. First, all of them will undergo transamination to cause these respective compounds: alpha keto isocaproic acid, alpha keto beta methyl valeric acid, alpha keto isovaleric acid, etc. And all of them will undergo branched chain keto acid dehydrogenase activity, which will be required in case of the activity of the keto acids. So now you will be requiring the dehydrogenase complex. So if this is deficient, you will be having maple syrup urine disease. Alpha keto isocaproic acid becomes isovaleric CoA. Alpha keto beta methyl valeric acid becomes alpha methyl butyryl CoA. Alpha keto isovaleric acid can become isobutyryl CoA. These are all components. Now then they will go for isovaleric CoA dehydrogenase. If this is deficient, sweaty feet syndrome can be seen. Then, if propenyl CoA has to become propenyl Coa Carboxylase, then if that enzyme is deficient, propionic acidemia can be seen. If propenyl CoA carboxylase is supposed to produce methylmalonyl CoA, and that methylmalonyl CoA has to become succinyl CoA, if that enzyme is deficient, you will be having methylmalonic acidemia. Did you understand this? If you are unable to see these words, please put your video on high definition. These are small letters, and put them on the big screen. You will be able to see them better. The logic is that let me erase everything that I have drawn here. I'm just telling you the common pathways through which branched chain amino acids can move forward. There is a disease called as maple syrup urine disease because of deficiency of branched chain keto acid dehydrogenase. In case of isovaleric CoA dehydrogenase, if there is a deficiency, you have a condition called as sweaty foot syndrome. And if propenyl CoA is trying to become succinyl CoA, I told you a pathway, right? We have a 3-carbon compound called as propenyl CoA trying to become a 4-compound called as D-methylmalonyl CoA. That will again rearrange to become L-methylmalonyl CoA. That will rearrange to become, you have succinyl CoA. In that process, if the propenyl CoA carboxylase is not acting, then you will be having propionic acidemia. That is also a disease. Okay. Okay. Identify the properties. It is not called as just mimicking. In this area, adenosyl B12, not mimicking at all. Adenosyl B12 is important for the enzyme methylmalonyl CoA mutase. Either if the enzyme is deficient or B12 is deficient, ADO-B12 is deficient, in both the conditions you can have methylmalonic acidemia, which can lead to methylmalonic acid urea. Now look at this question. Identify the properties descriptive of competitive inhibition. What is your answer? Identify the properties descriptive of competitive inhibition. Which one? Okay, Raman Thakur says 4, Prithvi Basurai says 4. What is 4? B and E. Excellent. So anybody else? Saurabh also says 4. Niranji Moon also says 4. What is 4? It is the presence of B and E. Remember, if this is the enzyme's active site, and if this is the substrate, and if this is the inhibitor, when inhibitor sits on the substrate, then the enzyme will not be able to function. This is called as competitive inhibition. When there is competitive inhibition, the attraction of the enzyme towards the substrate has decreased, because of which the Km value increases. I repeat, Km value is inversely proportional to the affinity of substrate to the active site. Km value is inversely proportional to the affinity of the substrate to the active site. Because somebody else is sitting in the active site, the enzyme is not anymore interested in the substrate, because of which affinity falls. So Km value increases. When affinity is falling, Km value increasing. But if I remove this enzyme by reversibility, if I increase the concentration of the substrate more and more, I'll be able to displace the inhibitor. When I try to flood the area with a lot of substrates, I'll be able to displace the inhibitor, then substrate can come and sit. The moment substrate is actually coming and setting, it remains the same kind of high velocity. So Vmax will remain constant. There is no decrease in the velocity maximum of the enzyme. So Vmax speaks about the enzyme's character. Vmax speaks about the enzyme's character. In competitive inhibition, is it reversible or irreversible? Come on, people. Are you understanding my question? I'm asking you, if an enzyme is like this, in the active site, substrate is coming and binding. If the inhibitor is there, if inhibitor comes and displaces the substrate, still the character of the enzyme is the same. But if this is the active site where the substrate may or may not bind, but there is an allosteric area where some inhibitor is coming and binding, because of which the enzyme's character has changed. Now it is no more capable of having its maximum action, then Vmax decreases. This is called as non-competitive inhibition. Sir, please, a negative allosteric activator inhibitor with examples. We don't have time for examples, but I can tell you the information here. Look at this part. See, if this is the enzyme and this is the active site, there is another site called as allosteric site. If a compound X comes and sits here, because of which enzyme action increases, it's called as allosteric activator. Allosteric activator. If Y comes and sits, because of which enzymatic activity decreases, it's called as allosteric inhibitor. If you want a better example for this situation, look at this part here. Glucose becomes glucose 6-phosphate. As it goes to the ultimate pathway, ultimately produces pyruvate. Now, this pyruvate is the end product of the whole glycolysis, which began with glucose. When more pyruvate is available, then glucose will not further go into the pathway. What is the enzyme that actually activates here? Yes, look at this part here. The first enzyme we have is hexokinase. So in glycolysis, we have three important enzymes. One is hexokinase, the second one is phosphofructokinase 1, the third one is pyruvate kinase. We have three enzymes which are irreversible. Why are they irreversible? Because they are not able to cross on the other side of the barriers of the energy. Also remember, these are very, very irreversible, because of which they become the bottleneck enzymes. Out of this, phosphofructokinase is actually the rate limiting enzyme. The meaning of the rate limiting enzyme is slightly different. In case of school, when you read about rate limiting reaction, it's different. Here it is different. Rate limiting enzyme means the enzyme on which maximum allosteric action can be noticed. For example, if insulin is present, that insulin will come and bind to the phosphofructokinase in a different area and can say the PFK can actually activate. But that is a very far ahead actual cause of thinking. Now imagine, if this is phosphofructokinase 1, where the fructose 1-phosphate, fructose 6-phosphate comes and sits, which becomes fructose 1,6-bisphosphate, then there is a phosphorylation happening. Now, you do not want any more fructose 1,6-bisphosphate to form if PFK1 is in action. So, the fructose 1,6-bisphosphate by itself can come and bind to an alternative site, because of which PFK1 cannot move forward. This is called as end product inhibition. Also, ATP, if it is in excess, it does not need further glycolysis. So ATP can also come and bind here. There is acting on a different site other than the active site. It can inhibit. Now imagine this. If I have insulin, insulin will actually activate, ATP will inhibit. Now if you have glucagon, glucagon will inhibit. These are all the different ways by which you will be able to understand. So these are the different ways by which you can understand how the allosteric activation has happened. Did you understand this? Superoxide dismutase, tell me, citrate will inhibit what? Why? Yes, that is a very good logic. Look at this part. If I start with glucose, ultimately pyruvate is formed, ultimately acetyl CoA is formed. Now this acetyl CoA has to enter into TCA cycle. When enough amount of glycolysis has happened, I have enough amount of ATP, I don't want any more glycolysis to happen. So when acetyl CoA is continuously entering into TCA cycle, there is more amount of ATP being generated. It means for every single cycle of TCA, you'll be treating 10 ATP. So acetyl CoA can go back and also inhibit phosphofructokinase 1 because further acetyl CoA formation can actually produce more amount of ATP which is not needed. Tell me how citrate is inhibiting. Do you know how citrate is acting? Listen carefully. Citrate will be a product of TCA cycle when there is excess amount of acetyl CoA. Now enough amount of glycolysis have happened, you have produced enough amount of ATP. At that point of time, you don't need more amount of TCA cycle. So the citrate will escape into the cytoplasm. Instead of entering the citrate, instead of becoming a part of TCA cycle, we focus on citrate which enters into cytoplasm. That citrate will go back and inhibit the PFK1 in such a way more amount of TCA cycle is needed, and at the same time ATP citrate lyase will come into action. That citrate lyase will break the citrate into again acetyl CoA and oxaloacetate. Now this acetyl CoA will travel into fatty acid synthesis or it can travel into cholesterol synthesis. So I repeat, ATP citrate lyase will be helpful in taking the citrate that has come into the cytoplasm and push the citrate into breaking down into acetyl CoA and oxaloacetate. That acetyl CoA and oxaloacetate, acetyl CoA will enter into fatty acid synthesis. Next question. Mark the enzymes that get activated by covalent modification called phosphorylation. What is your answer? Thank you, Prithvi Basurai. Tell me, what is the answer for this question? Okay, Ibiza says B, Prithvi Basu says C, Himalini says C, Ashwardhan says C, Saurabh says D, Superoxide dismutase says C, Raman Thakur says A. Excellent, the answer is 3,4, which is C. So the answer is this one. Now remember, there is something called as covalent modification. If you look at the theory part of the enzymology, the enzyme regulation can be done in 3 different ways. One is called as activation or inhibition. This can be done mechanically or by allosteric activation. The next thing is by induction or repression. If I say activation inhibition, that is actually controlling the activity of the enzyme. When I say induction repression, induction means the genes are getting transcribed and they are becoming translated to produce enzymes. This speaks about the quantity of the enzymes, not the activity, the quantity of the enzyme. And the third one will be covalent modification. This covalent modification is reversible, sometimes it can be irreversible, and that again speaks about the activity of the enzyme. For example, if I think about glycogen formation from that of glucose. Glucose becomes glycogen, glycogen becomes glucose. When glucose is becoming glycogen, it is glycogen synthesis. The most important enzyme is glycogen synthase, the rate limiting enzyme. If glycogen is breaking down into glucose, it is glycogen phosphorylase, which is the rate limiting enzyme for breakdown of glycogen into glucose. Now glycogen synthase will be active if the phosphate is removed from the glycogen synthase, while glycogen phosphorylase is active if it is phosphorylated. On phosphorylation, glycogen phosphorylase is active. On phosphorylation, glycogen synthase is inactive. That is how you will be able to control the opposite pathways. This is what you are supposed to look for. Now look at this question here. There are examples of enzymes who can be activated on phosphorylation. It is citrate lyase, that is ATP citrate lyase, glycogen phosphorylase, phosphorylase B kinase, HMG-CoA reductase kinase. These are the most common enzymes where you will be looking for catabolism. Generally, catabolism can be seen. In case of citrate lyase, anabolism can be seen. Why? Because I told you ATP citrate lyase is able to break the citrate to produce acetyl CoA, and that acetyl CoA can travel into the formation of fatty acid. Okay. Now here also you're looking for anabolism, where HMG-CoA reductase kinase means the kinase is capable of causing phosphorylation of HMG-CoA reductase. If HMG-CoA reductase is activated, then cholesterol synthesis happens. So mark my words. In the given 4 example, the first example and the fourth example are examples of phosphorylation helping in anabolism. These two are examples of glycogen breakdown because catabolism is being activated by phosphorylation. I hope you will be able to remember this in your exams. Okay, what is the answer for this question? This is a previous year question. Identify the common thread statement in all serine proteases. Identify the statement that acts as a common thread in all serine proteases. Superoxide dismutase says it is 3. Anybody else? Bizarre says 4. Anybody else? Prithvi Basu Roy says 2. Okay, so we'll be doing the remaining questions faster and faster because we started exactly at 7 o'clock. It's going to be 9 o'clock. We'll be wrapping up the class by 10 o'clock. In 1 hour, we'll finish almost 60 questions, I believe. So please be with me. Tell me faster the answer. Okay. See, serine protease. Do you know what is the meaning of serine protease? When I say aspartyl protease, the meaning of that is you are looking at an enzyme called as protease, which is going to break a protein. You're looking at an enzyme called as protease that breaks the protein. Now, in that active site, you have serine. If the enzyme has serine in its active state, that is called as serine protease. If the enzyme has aspartate in the active state, it is called as aspartate protease. So when they say serine protease, in the active site, you have serine, histidine, aspartate as the catalytic triad at the active site. This is the true statement. On the other side, nothing else is true. There is no tight binding of pancreatic trypsin inhibitor, wrong. And there is no autocatalytic activation of zymogen precursor. If there is autocatalytic activation of zymogen precursor in the pancreas, that is the reason for pancreatitis. This is not physiological. Okay, have a look at this. There are serine proteases because they hydrolyze proteins and they have essential serine residues at the active site, which is critical for catalysis. Who are the best examples of serine protease? Trypsin-like, chymotrypsin-like, thrombin-like, elastase-like, subtilisin-like. Okay, take a note of this. Trypsin-like, chymotrypsin-like, subtilisin-like, elastase-like, thrombin-like. 5 different types of, why not 2? Cleavage protein on the carboxyl site of serine residues. There is no guarantee that it has to be on the serine residues. They are looking at the word serine, right? It is to confuse you. It can be at any residues. Serine protease does not mean it is actually having predilection for the serine side. Even if you have glutamate here, if you're trying to cleave it from the carboxylic side, still it can happen. Just because it is a serine protease, it does not show you any kind of special predilection for the serine residues. Did you understand this? Read the question here. Cleavage of a protein on the carboxylic site, of course understandable, but not from the serine side. Okay, next question. Look at this question here. This is a lengthy question. If you get these kind of questions, you should be ready for reading them faster to produce an answer. Child with abnormally shaped and large cranium also presents with short and malformed extremities in the limbs. She has a unique blue sclera. X-ray findings suggest multiple limb fractures. There is a significant reduction in the amount of type 1 collagen in the baby's skin. DNA analysis demonstrates the presence of 2 normal alpha 1 alleles and 1 normal alpha 2 allele. Which of the following statements best describes the given clinical picture? First, tell me, what is the clinical picture? What is the diagnosis? What is the diagnosis? Excellent. This is a case of osteogenesis imperfecta. When you know it is osteogenesis imperfecta, among the options A to E, which one exactly fits it? Which one fits? You're welcome, classes fan. Thank you. Osteogenesis imperfecta is not about the alpha 1 collagen peptide synthesis deficiency. You can see the presence or reduction in the amount of type 1 collagen in the baby skin. But type 1 collagen is not the only problem in this area. Inability of alpha 1 to form triple helix, defective alpha 1 chain interrupting triple helix formation, none of them are a problem. The problem is option D, incorporation of defective alpha 2 chains that cause instability and degradation of the triple helix. I repeat, the alpha 1 collagen will be synthesized even though it is lesser, and after synthesis triple helix also will be formed even though it is lesser. But after the formation of triple helix, you will be able to have the destruction of the triple helix because of the defective alpha 2 chains. So I'll put them in simple words. The alpha 2 chains will be wrongly or improperly incorporated. If they are improperly incorporated, then they can destroy or disrupt the already formed triple helix. This is what I want you to know. Did you understand this, people? Did you understand this? Hello, can I go to the next question? Okay, pick up the correct statement about isoenzymes. Pick up the correct statement about isoenzymes. They have the same quaternary structure, they have same distribution in different organs, same enzyme classification with same enzyme number and name, catalyze the same reaction. Which one is the true statement here? This is a very commonly asked MCQ. Whatever MCQs I have actually told you, they have been slightly modified from the previous year questions. So please be with me if you think you can answer them better. Okay. Superoxide dismutase says 4, Ibiza says 4, Prithvi Basu says 4. Very good. Almost all of them are getting the right answer. The right answer is option 4. Remember, isoenzymes, for example, LDH can be of 5 types. 1, 2, 3, 4, 5. And in case of CK, creatine kinase, you can have 3 types. MM, MB, BB. It means you are getting a quaternary structure based on the multiple subunits that you are using. A subunit is something who will be referred to as a tertiary structure. And if at all I'm going to bring 2 subunits together, that automatically becomes a quaternary structure. So in case of CKMM, 2 subunits are MM. In case of BB, there are completely different types of subunits. So same quaternary structure is wrong. And CKMM is mostly present in the skeletal muscle, while MB is present in the normal, Yes, MB is present in the cardiac muscle and BB is present in the brain. So they cannot be having the same distribution. And so whenever you have a different kind of isoenzymes, you will be having a different enzyme classification number. But a creatine kinase will perform the same reaction where creatine is converted into creatine phosphate in the presence of ATP that becomes ADP. This will be again reversed back. Creatine phosphate becomes creatine where ADP becomes ATP. Can you tell me what is the name of the reaction? Creatine kinase is capable of helping the conversion of creatine to creatine phosphate, and the ATP becomes ADP. And when the creatine phosphate becomes creatine, ADP becomes ATP. Can you tell me the name of the reaction? The name of the reaction is Lohmann reaction. Lohmann reaction. Please remember option 4 is the right answer. Okay, what are the points? Isoenzymes catalyze the same reaction. The different quaternary structure, tissue distribution is different. Enzyme name and number can be different. It is not Lohmann, it is Lohmann, Lohmann. Okay, the enzymes mannose 2, N-acetylated glucose transferase 1, galactosyl transferase are all markers of what? Just give me a moment. Yes, excellent. They all belong to Golgi apparatus. That is your right answer. Very good, people. You have got the right answer. Yes, it is meant for Golgi apparatus. Yes, most of you have gotten the right answer. It is Golgi apparatus. So now this is an area where you will be having multitudes of MCQs coming to you. What is the marker for plasma membrane? You will be looking for the membrane, that is peripheral proteins that can be present on the plasma membrane. You have to learn about the markers of each organelle. In case of what is microsome? Microsomes are nothing but the broken down endoplasmic reticulum after the proper centrifugation reaction. If you see them in vivo, you call them as endoplasmic reticulum. If you see them in vitro, you call them as microsomes. What is the marker for mitochondria? The TCA cycle enzymes will act as the marker for the matrix. The electron transport chain components can act as the markers for the mitochondria. Okay. Yes, for endoplasmic reticulum, it is glucose 6-phosphatase. Enzymes which are raised in liver disorder is RR. What is your answer? Cis end, trans end. Look at this. When I have a nucleus here, the nuclear membrane will expand like this, and you will be having them as ER. Now, when the proteins are formed in the ER, it means that ER is containing a lot of ribosomes, making it as an RER. And the protein synthesized will be released as vesicles. Those vesicles are fresh peptides. And those fresh peptides will come and touch the Golgi apparatus. This Golgi apparatus will be seen like this. And now, when it's touching this one area, that portion of the Golgi apparatus which is facing the nucleus will be called as cis Golgi. And once they are traveling into the Golgi area, they'll be traveling between this part. This portion is called as trans area in the opposite side. When they come out of this particular side, they will be not just peptides, they'll be proper folded proteins. Proper folded proteins. So you have the side facing the nucleus referred to as cis Golgi, and the other one that is opposite to that would be called as trans Golgi. Between that is called as trans Golgi network. Is that clear? Saurabh Banashali, have I explained to you the meaning and the difference between the two? The Golgi apparatus, which is receiving the fresh peptides from the nucleus and the RER will be called as. Glycosylation can happen anywhere. It is happening through the cisternae. When they are traveling through the cisternae, the addition of glucose, mannose or any form of sugar can happen. The addition of sugar will be referred to as glycosylation. They happen in the cisternae. Oh, ALP is repeated twice. I'm so sorry. Okay, you know, if they are looking at the liver disorder, it will be ALP. This is supposed to be ALT. I'm so sorry. ALP, ALT, AST. The answer should be 3, 4, 5. The answer is C. Okay. Phase 2 reaction. You are asking me, see, you are asking me questions that can take me into a different direction. Still, let me answer you. There is something called as biotransformation. Biotransformation is the name that you give for a chapter called as xenobiotics. Here you have to realize xenos means alien or also called as foreign. If at all you take any kind of chemical compound into your body, which is a foreign compound, your body has to metabolize it. So there will be a transformation happening biochemically, because of which a toxic compound is converted into a non-toxic compound. It is technically a insoluble compound is converted into a soluble compound, because of which it can become more soluble in water, through which it can be lost in the urine. There is the ultimate process of biotransformation. I have told you about biotransformation in the shortest way possible. Biotransformation means if at all you are going for a biochemical metabolic pathway, you are chemically trying to convert a non-toxic compound, toxic compound into non-toxic compound. How? Because you are trying to convert an insoluble compound into soluble. It becomes soluble in water. It is lost in the urine. This is done. Now it will be happening in two phases. In phase 1, you undergo simple chemical reactions like hydroxylation reactions, oxidation reaction, reduction reaction, hydrolysis, etc. But phase 2 reactions will be called as conjugation reactions. It means you're trying to add a very special compound to the molecule so that you make it more soluble. That is, if you go for a glycine addition, for example, benzoic acid plus glycine will give you a compound called as hippuric acid. Then you will be having glucuronic acid. Glucuronic acid is added to bile bilirubin, because of which you can have bilirubin monoglucuronide, bilirubin diglucuronide. Likewise, you can have sulfation, you can have methylation. All these things can be taken as addition of a special compound through which you will be able to release an organ. Now the special compound addition is called as phase 2 reaction. Okay, what is the answer to this question? Key glycolytic enzymes are all except which one? Prithvi says C, Himalini says C. Anybody else? Excellent. So who are the key glycolytic enzymes? This is a key glycolytic enzyme because this is the rate limiting enzyme itself. Hexokinase is also an irreversible enzyme. This also belongs to glycolysis. Pyruvate kinase is the last enzyme in the pathway. That is also glycolytic enzyme. But glucose 1,6-bisphosphatase never exists. Instead, you can have fructose 1,6-bisphosphatase. That belongs to gluconeogenesis. Okay, it belongs to gluconeogenesis. So the only one that does not belong to the glycolysis would be option 4. Option 4 is option C. Okay, answer. Cancer cells derive nutrition from which pathway or which method? Cancer cells derive nutrition. How? Superoxide dismutase says 2. Anybody else? Himalini says 2. Anybody else? Any other answer? Padmaja says 2. Prithvi Basu says 2. Excellent. So the answer is glycolysis, obviously, because even if it's a cancer cell or a microbe, protozoa, prokaryote, whatever it is, the universal source of nutrition will always be glycolysis. I mean, universal source of energy will be glycolysis. And that glycolysis in cancer cells will be explained by what hypothesis? It is explained by Warburg hypothesis. Let me tell you what is Warburg hypothesis. Cancer cells' primary source of nourishment is through glycolysis, obviously. What does it say? The increased ratio of glycolysis to aerobic respiration was likely due to defects in the mitochondrial respiratory chain. The second thing is, enhanced glycolysis enabled cancer cells to preferentially proliferate in the reduced oxygen tension often seen in tumors. Switch from aerobic to anaerobic glycolysis propels tumor formation, growth and propagation. So what does he say? If the tumor cells are present for X area of place where Y number of cells are supposed to be present, if there is a tumor, let me repeat once again, for the X space available, X nutrition available, X vasculature available, normally if Y number of cells are present. But suddenly, if 20 Y cells are present for the same nutrition, then you will not be able to continue with aerobic glycolysis because the amount of cells have increased massively, but the available nutrition is lesser. So you have to switch towards anaerobic glycolysis. In anaerobic glycolysis, you will be converting pyruvate into lactate. So the enzyme active will be LDH. That is why in any kind of tumor, a non-specific marker can be LDH elevation in case of tumor. And in spite of that, growth and propagation happens. So remember, mitochondrial respiratory chain will not be happening because you will be going for anaerobic glycolysis. Next question. Enzyme responsible for complete oxidation of glucose to carbon dioxide and water is present where? Come on, what is the answer of this question? What is the answer of this question? Enzyme responsible for complete oxidation of glucose and carbon dioxide and water is present where? Padmaja says 2. Anybody else? Vaishali says 2. Prithvi says 4. He says lysosomes. Saurabh says 2. Okay, let me make it very simple. Complete oxidation. Complete oxidation is the word you use for a pathway called as TCA cycle. Direct oxidative pathway is the word you use for HMP shunt. Direct oxidative pathway is the word you use for HMP shunt. So when there is a complete oxidation, that is glucose is completely converted into carbon dioxide and water, that happens in TCA cycle. It happens in the matrix of mitochondria. Okay. Okay. Single base pair substitution is related to which of the following? Among the investigations we run, single base pair substitution is related to which of the following? Excellent. It is restriction, fragment, length, polymorphism. You are trying to look for polymorphism happening over a small area in a community. There are people like 1, 2, 3, 4, 5, 7, 8, 9, 10, 20 people are there. Out of them, in a single small space in the whole of genetic information, you're looking for how each human being can differ. That is polymorphism. And when you are doing it, you're using restriction enzymes because of which you're making fragments of different lengths. Here, you will be able to look for a single nucleotide being changed. If, for example, A has been replaced by T, you will be able to understand the pair substitution has been done. But what is PCR? PCR is a way by which a nucleic acid can be amplified. A nucleic acid can be amplified with the help of polymerase chain reaction into millions and millions of times. Southern blot is a method of identifying the presence of a specific DNA using DNA probe. For example, X is the DNA that you are looking for, and in the particular test sample you have a DNA. You want to see whether this DNA and this DNA are the same, then you try to blot them. When you blot them, you're able to identify both of them are similar, then it has been understood as the same. FISH is fluorescent in situ hybridization. If you find a particular chromosome, a specific chromosome or chromosomal region because of hybridization, and then you add a fluorescently labeled DNA, and when there is a glow, you will be able to identify that that chromosome is present. So term RFLP is the only one that refers to a single base pair substitution in the germline DNA of different individuals that either destroys or creates a new recognition sites for a given restriction enzyme. Mitochondria is involved in which pathway? Come on, tell me, what is the answer for this question? Tamogna says 5. What is 5? Vaishali says B. It is 2, 3 and 4. Anything else? So Vaishali's answer is the excellent answer, because fatty acid synthesis always happens in the cytoplasm. Remember, cytoplasm is known for reduction and also for biosynthesis, and generally for anabolism. When it comes to mitochondria, it is generally done for catabolism. On that process, remember, mitochondria has its own DNA. With the help of mitochondria DNA, you can go for DNA replication also. And fatty acid oxidation, called as beta oxidation of fatty acids, happens in the mitochondria. And from the DNA of the mitochondria, the protein mitochondrial proteins also can be synthesized. So 2, 3 and 4 is the right answer, except fatty acid synthesis. Everything else is right. Now let's go for this question. Acetyl Coa Carboxylase is not activated by? Yes. Acetyl Coa Carboxylase is not activated by? What is your answer? Remember the logic. When you start with acetyl CoA as the substrate, which is very small molecule, you keep on building the molecule to produce something called as palmitic acid or palmitoyl CoA. You can produce this. This is the ultimate product. In that process, acetyl CoA carboxylase can be a rate limiting enzyme. Now this is not activated by, look at this, acetyl CoA can activate acetyl CoA carboxylase because the reaction can move forward. But the instant step of acetyl CoA carboxylase is malonyl CoA. Malonyl CoA is the substrate, is the product of acetyl CoA getting carboxylated. So malonyl CoA will only inhibit the acetyl CoA carboxylase. So it is not going to be activated by malonyl CoA. At the same time, citrate. Citrate, its function is to break into acetyl CoA and that of oxaloacetate. So when you can give acetyl CoA, that acetyl CoA will be activating the acetyl CoA carboxylase. So this is a true statement. And palmitoyl CoA will ultimately be the last end product. The end product inhibition is what you say. Palmitoyl CoA can come back and inhibit the acetyl CoA carboxylase. So this can be taken into consideration. Or about acetoacetate, that is where the confusion lies. So 1, 3, 4 can be the right answer. Acetoacetate is the product of acetoacetate is the product of ketone body synthesis. How did the ketone body synthesis happen? When the acetyl CoA molecules have been converted into acetoacetate. Now this acetoacetate may be inhibiting the acetyl CoA from entering into the TCA cycle, and entering into the ketone bodies. Acetoacetate is entering into ketone bodies, right? What are the important enzymes? You have HMG CoA synthase and HMG CoA lyase. These are the two enzymes important in the ketone body synthesis. So acetoacetate may inhibit the HMG CoA lyase. but at the same time it is also capable of inhibiting the acetyl CoA carboxylase because it wants all the acetoacetate to enter into the palmitoyl CoA pathway. So malonyl CoA, palmitoyl CoA, acetoacetate all can inhibit the acetyl CoA carboxylase, while citrate will activate acetyl CoA carboxylase. Again, why? Because citrate bakes into acetyl CoA and oxaloacetate. Acetyl CoA has only one pathway to move forward if there is anabolism to form fatty acid. When there is a fatty acid synthesis going on, then citrate will activate acetyl CoA carboxylase. Bradford assay is done in laboratories for estimation of what? Bradford assay is done in the laboratories for the estimation of what? Anybody? A small request to all of you. Generally, people are not in a mood to listen for any MCQ sessions or directly any other live sessions because they'll be seriously studying with their books and preparation. You are the people who have been kind enough to come for live classes and sit and listen to what a teacher is wanting to say. So I respect your bravery and your goodwill. And if you have chance, please put a thumbs up after the class is over. This thumbs up can help the video stay alive to reach more people. For that reason, please put a thumbs up at the end of the session. Okay, Bradford assay is done for. See, Bradford assay is not done for glucose because glucose estimation can be done with the help of hexokinase method, NADH method. It can be by glucose 6-phosphate dehydrogenase method. It can be glucose oxidase method. It can be done by isotope dilution mass spectrometry. I repeat, hexokinase method, glucose 6-phosphate dehydrogenase method, glucose oxidase method, isotope dilution mass spectrometry, and glucose can be identified by Benedict's test. Creatinine is estimated by kinetic Yaphase test. Urea can be estimated by TMX DOA test. Okay, thiosemicarbazide test can be done in case of urea. Proteins are generally estimated by Biuret test, but also by Bradford assay. Bradford assay. This is not done for peptides, not done for glucose, not for creatine, not for urea. The Bradford assay is done for proteins. This is a dye binding assay and the dye would be Kumasi brilliant blue. After the change in color, you will be able to estimate using colorimetry or spectrophotometry. Remember, it's a dye binding assay which uses Kumasi brilliant blue. Bradford assay is for proteins. Next question. Maximum reduction in HbA1c is caused by which of the following drugs? Maximum reduction in HbA1c. For example, if a patient is suffering from either type 1 diabetes or type 2 diabetes. Most common people are type 2 diabetes. 85% population is type 2, only 15% population is type 1. In this patients, the HbA1c can be used as a tracker for the treatment and control of diabetes. So for that you will be giving multiple oral hypoglycemic agents, even you will be giving insulin. Among them, the maximum reduction in HbA1c can be caused by which drug? Okay, Prithvi Basu is saying C, which is biguanides. Albatross is saying B, which is thiazolidinediones. Padmaja is also saying thiazolidinediones. Any other answer? Himalini says B. I'm really sorry. You should not be saying this answer. The answer is sulfonylureas. Please remember, nobody can beat sulfonylureas in being the maximum reducers of HbA1c. Sulfonylureas can be helpful for what kind of diabetes? Can you tell me? Use the logic and tell me. See, type 2 diabetes is generally considered as insulin non-dependent. But you're not supposed to use the non-insulin dependent as the word. Type 2 diabetes can be of two types, A and B. A is a condition where there is insulin resistance. Insulin is available, but the end organs are resistant towards the insulin activity. Type 2B is poor insulin secretion. Poor insulin secretion from that of the beta cells of pancreas. If you take into consideration any other oral hypoglycemic drug, they are trying to bring the glucose down by multiple mechanisms. But when you look at sulfonylureas, the sulfonylureas will act as secretagogues and they are capable of releasing the insulin itself. Nobody can decrease the HbA1c like insulin. So thiazolidinediones can be easily beaten by the sulfonylureas. Did you understand this, people? So maximum reduction of HbA1c. Maximum reduction in HbA1c is caused by sulfonylureas, while the minimum reduction of HbA1c is caused by alpha glucosidase inhibitors like miglytalan acarbose. Miglytalan acarbose. Okay. Next question. Which of the following vitamin deficiency can simulate a picture of orotic acid urea? Which of the following vitamin deficiency can simulate a picture of orotic acid urea? What is your answer? Yes, it is vitamin B12. When there is vitamin B12 deficiency, the patient will suffer from megaloblastic anemia. The megaloblastic anemia means you will be having macrocytes and megaloblasts with a huge RBC red cell distribution with being higher. Different kinds of cells can be seen. And the hemoglobin concentration may be lesser, the content will be the same. Now B12 deficiency will look like orotic acid urea because orotic acid urea happens because of pyrimidine synthesis pathway defect. There is a defect in the pyrimidine synthesis pathway. Here also you will be having megaloblastic anemia as the picture. Why? Look at this part. In orotic acid urea, there is an error of merbendum metabolism. You will be having retarded growth, development, megaloblastic anemia, unresponsive to B12 deficiency. And why is that? Because it is due to decreased pyrimidine synthesis, which leads to decreased nucleotide lipid cofactors needed to erythrocyte membrane synthesis in the bone marrow. I repeat, if pyrimidine synthesis is lesser, no Prithvi Basu, CPS2 is the one that is involved in the pyrimidine synthesis, but when there is a CPS1 deficiency, then you will not be able to produce CP at all, so that CP will not be able to fall into the cytoplasm. Okay, you have pyrimidine synthesis having defect. Because of that, you will be having decreased nucleotide lipid cofactors needed for erythrocyte membrane synthesis in the bone marrow. If the membrane synthesis of the RBCs are not happening in the bone marrow, megaloblastic cells can happen. Okay, amino acid substitute for tryptophan without altering protein characteristics is which one? Name the amino acid substitute for the amino acid tryptophan without altering the protein's characteristics. What is your answer? Himalini says C. Anybody else? The first and foremost logic is tryptophan is an aromatic amino acid. So you look at methionine, it is aliphatic. Look at alanine, aliphatic. Tyrosine, phenylalanine, both are aromatic. But what is tyrosine? Hydroxy version of phenylalanine is called as tyrosine. Tyrosine is hydroxy version of phenylalanine. If there is a hydroxylation, then it actually has more amount of polarity compared to that of other amino acid. But when it comes to phenylalanine, this is also a pure aromatic amino acid. This is also an aromatic amino acid. So phenylalanine can act as a substitute for tryptophan without altering the protein's basic physical characteristics. Is that clear? Yes. Now let's go for the next question. Statins are nocturnally administered drugs because why? The statins are given, they are actually capable of inhibiting HMG coreactase in cholesterol synthesis pathway. Statins are given in the night. Why? Statins are given in the night. Why? Rosuvastatins and related statins are exceptional because they can be given any time of the day because they have 24/7 action. But generally, HMG coreactase activity is maximum in the night. Why? Because whenever you are having a daytime activity, during the daytime, the kind of food you eat is a fuel. It can be used for physical activity. So you will be burning the fuel. But as it progresses towards night, in the night insulin becomes active, because insulin will try to utilize all the fuel which was not utilized and tries to store the fuel into either fats or proteins. So anabolism starts in the night. And one of the ways by which insulin can activate anabolism is cholesterol synthesis and also fatty acid synthesis. The rate limiting enzyme of fatty acid synthesis called as acetyl Coa Carboxylase or methionine synthase, and cholesterol's rate limiting enzyme called as HMG-CoA reductase, they both will be maximally active in the night. So because of that, when you give a statin in the night, it will have maximum effect. I hope you have understood the reason behind it. Next question. Quarter staggered arrangement is a term used in describing what? Mucopolysaccharides and the structures, structure of collagen, structure and function of elastin, only the function of desmosin. Which one? Anybody? Yes, superoxide says B. Anybody else? Shaurabh says B. Yes, quarter staggered arrangement is the word you use to describe the structure of collagen, where the triple helix has to be strengthened. Strengthening of triple helix can be done with the help of quarter staggered arrangement. It increases the tensile strength of collagen. Increases the tensile strength of collagen. Look at this part here. These are the collagen fibrils. You have multiple microns. Now they will be present somewhere here. Now if you look at this part, the collagen microfibrils can be understood here. And these are the collagen molecules as such. Alpha 2 helix, alpha 1 helices. You mean you will be having two alpha 1 helix and one alpha 2 helix coming together. Now this portion is called as quarter staggered arrangement. What is the meaning of it? One particular collagen microfibril will be here, one will be here, one will be here, one will be here. It means there is a 25% gap here, 25% gap here, 25% gap here. That particular gap will be arranged in such a way, here this one and this one will be facing each other, this and this will be facing each other, this and this will be facing each other. So when there is a pull, not all the fibers will come together, all the four will be happening in a slanting direction, because of which the strength of pull will be higher. So the whole tensile strength of the collagen fibers will be bigger, which is because of quarter staggered arrangement. Let me draw it again. One fiber will be here, another fiber will be here, another fiber will be here, another fiber will be here. It looks as if somebody has drawn steps here, and this will be 25% gap, 25% gap, 25% gap. This is called as quarter staggered arrangement. There is a stagger. Stagger means what? There is a space being given. Staggered arrangement. And that is the explanation for the collagen structure. Next. It is lateral association of the triple helix units. Okay. Identify the collagen involved in osteogenesis imperfecta and Ehlers-Danlos syndrome type 7 EDS. What is the collagen involved in osteogenesis imperfecta and Ehlers-Danlos syndrome type 7? Remember, be it type 7 Ehlers-Danlos syndrome or osteogenesis imperfecta, osteoporosis, the answer will always be involved in the type 1 collagen A1. Call A1, call 1 A2. Osteogenesis imperfecta, osteoporosis, Ehlers-Danlos syndrome, subtype arthrochalasia. You can also remember collagen 2 is important for severe chondrodysplasia, collagen 3 is for regular Ehlers-Danlos syndrome, and collagen 4 is for Alport syndrome attacking your kidney, sometimes in the lung. Collagen 7, epidermolysis bullosa and dystrophic property. We'll repeat once again. Collagen 1, osteogenesis imperfecta, Ehlers-Danlos syndrome. Collagen 2, severe chondrodysplasia. Collagen 3, Ehlers-Danlos syndrome with vascular subtype. Collagen 4, Alport syndrome. Collagen 7, epidermolysis bullosa. Okay. Glucocorticoids which can retain maximum salt is which one? The glucocorticoid that can retain the maximum salt. It means which property are we looking for? In a glucocorticosteroid, which property are we looking for? When we speak about retaining maximum salt, which property, salt means sodium, okay? Which property are you looking for in a glucocorticoid? Okay, Prithvi says D. Anybody else? I'm asking you, what is the meaning of the question? Glucocorticoid which can retain maximum salt means what are they looking for? They are looking for the mineralocorticoid property. Remember, all glucocorticoids can have at least minimal amount of mineralocorticoid property. They're asking you, glucocorticoid with the maximum mineralocorticoid activity is which one? The answer is hydrocortisone. Now, let me tell you a few important properties here. The glucocorticoid which has the minimum glucocorticoid activity is hydrocortisone, because hydrocortisone has the maximum mineralocorticoid activity. I repeat, glucocorticoid with the minimum glucocorticoid activity but maximum mineralocorticoid activity is hydrocortisone. The maximum glucocorticoid activity ever seen is seen in dexamethasone and betamethasone. That is, the steroids which have maximum glucocorticoid activity is dexamethasone and betamethasone. Longest acting glucocorticoid is dexamethasone, and the longest most acting would be triamcinolone. Mineralocorticoid with no glucocorticoid activity at all is deoxycorticosterone acetate. The mineralocorticoid who will never have any glucocorticoid activity is deoxycorticosterone acetate. The most potent mineralocorticoid is aldosterone. Is that clear? The least potent glucocorticoid is cortisone. Okay, let's move forward. Hartnup disease causes deficiency of what? Which ones to remember? You have to remember all of them. See, you will be knowing glucocorticoids which will have mineralocorticoids also. Now, the glucocorticoids will be having glucocorticoid activity also. Which glucocorticoid has the most glucocorticoid activity? It is dexamethasone. And who has the longest duration of action? Triamcinolone. For example, if there is any kind of ligamental injury where you want to have a steroid being placed inside, triamcinolone will have maximum effect for a longer time. Mineralocorticoid with no glucocorticoid activity will be deoxycorticosterone acetate. The potent mineralocorticoid is aldosterone. All these are basic informations. Hartnup disease causes deficiency of, yes, nicotinamide. What is Hartnup disease? It is a defective transporter of neutral amino acids in which tryptophan reabsorption in the kidney is failing. So you keep on losing tryptophan in the urine. Tryptophan is lost in the urine. You people remember, tryptophan is the source of another vitamin called as niacin. So tryptophan has to be helpful for the synthesis of vitamin B3. Tryptophan is essential for vitamin B3 synthesis. But for the whole pathway, you require vitamin B6. So Hartnup disease is a disease where tryptophan reabsorption fails, and so because of deficient tryptophan, niacin is not synthesized, because of which pellagra-like symptoms can be noticed. Pellagra-like symptoms can be noticed. Next, try to link these areas. Try to link these two. The diseases on the left side, channels involved on the right side. Hyperkalemic periodic paralysis, sodium channel. Hypokalemic periodic paralysis, slow calcium voltage gated channels, DHPR. Malignant hyperthermia, calcium release channel, RYR1. Myotonia congenita, chloride channel. Which one? How do you compare? Is it 1B, 2C, 3D, 4A, 1A, 2C, 3B, 4D, 1A, 2B, 3C, 4D, 1D, 2A, 3B, 4C? Which one? Is it A, B, C, D? Which one? What is your answer? So don't worry much about it. This is a teaching MCQ where the question is a googly question. There is no difficulty in this. 1 is for A, 2 is for B, 3 is for C, and 4 is for D. Please remember them. Hyperkalemic periodic paralysis is involving sodium, while hypokalemic periodic paralysis involves slow calcium, which is a voltage gated channel. When it's malignant hyperthermia, it is calcium release channel from the RYR, ryanodine receptor. Ryanodine receptor. If at all you focused on the option 3 only. 3 is for C. Where do you get 3 for C? Here you have 3 for D, 3 for B, 3 for B. So it means there is only one option which has 3 for C. You could have easily cracked C if you know 1. Myotonia congenita or congenital myotonic dystrophy involves chloride channel. So remember, hyperkalemic periodic paralysis or hypokalemic periodic paralysis, both of them are not involved with potassium channel. Both of them are not involved in potassium channel. The hyperkalemic is involved in sodium channel and hypokalemic is involved with slow calcium channel, while myotonia is with chloride channel. Malignant hyperthermia is in case of calcium release channel. That's all. Is that clear? So you can look at this later. Now tell me the answer of this question. Okay, let's not do this question. Okay, let's do this question. Among the following, which is an extracellular event in collagen synthesis, I have focused much on these tough areas because these are the areas where MCQs have been asked left and right. So I want you to be prepared for this. Tell me what is the answer for this. Collagen synthesis will have two types of events: extracellular event, intracellular event. In collagen, there have been totally 84 MCQs in the last years between 1995 to 2015. Between these two, 84 MCQs have been asked in the national exams. In case of INICT types of exams like JIPMER, PJ Chandigarh, etc., approximately another 120 MCQs have been asked. So totally 200 MCQs have been asked approximately in the past 20 years. So you should be knowing them. Okay. So you have collagen synthesis divided into two parts. One is extracellular, the other one is intracellular. Look at this. Assembly of collagen fibers in quarter staggered alignment is an example of extracellular event. When I spoke about a previous question, quarter staggered arrangement is an extracellular event. They're asking you among the following which is an extracellular event. Assembly of collagen fibers in quarter staggered arrangement is an extracellular event. Who are the other extracellular events? If you have cleavage of amino carboxy terminals for propeptides, oxidative deamination of amino groups like lysyl and hydroxylysyl residues of aldehydes, formation of intra and interchain linkages using Skiff's bases, they all are extracellular activities. Who are the intracellular events? Cleavage of signal peptide, hydroxylation of prolyl residues and lysyl residues, glycosylation of some hydroxylysyl residues, formation of intrachain interchain bonds and extension peptides, they all are intracellular events. Remember, hydroxylation of prolyl residues and lysyl residues are examples of intracellular events. Okay, next question. Reduction of glucose with calcium in water produces what? Reduction of glucose with calcium in water produces what? Reduction of glucose. Sugar, alcohol. What is it? Superoxide dismutase among them? Excellent. So the answer is A. If at all you reduce galactose, that will produce galactitol, which is called as dulcitol. Mannitol is not coming from here. Glucuronic acid happens because of oxidation, not because of reduction. So the answer is sorbitol. Okay. Next question. Which of the following is the significant adverse effect of rifampicin as it acts as an RNA polymerase inhibitor? When you look at it as a molecular biology question, that is an RNA polymerase inhibitor. Tell me, what can be the significant adverse effect? Yes, because liver is the most important organ for all metabolic pathway, excretory mechanism, secretory, synthetic, storage activity, and homeostatic mechanism. Metabolic means all carbohydrate, lipids, proteins, nucleic acid metabolism. Excretory means bile. Secretory means many proteins. Synthetic means synthesizing many proteins including albumin and factors 2, 7, 9, 10. And storage of glycogen and other kind of storage disorders. Homeostatic by maintaining the oncotic pressure. All these are functions of liver. So liver has to be continuously replicating. In the replication, RNA polymerase is important. If the RNA polymerase is blocked by the rifampicin, then hepatotoxicity will be the most important significant adverse effect. The other adverse effects you will be knowing later. Reducing ability of carbohydrates is due to what? Tell me. The reducing ability of carbohydrates is due to what? Yes. Anybody? Okay, remember, generally the reducing ability of carbohydrates is due to the carbonyl group. That is either the CHO present in the aldo sugar or the C double bond O keto group present in the keto sugar. Example of aldo sugars can be your glucose, glucose, mannose, galactose, etc. Keto sugars example can be fructose. But even though you have a carbonyl carbon, only when they form enediol. What is enediol? You have an OH here, you have an OH here. Between the two carbons you have a double bond. This is called as enediol. Enediol formation is the most important requirement for the carbohydrates to act as a reducing sugar. Next, among the following hormones, which one gets activated in the liver? What is the answer for this question? Among the following hormones, which one gets activated in the liver? Come on, people. This is an intelligent question. It takes efforts to answer this. I'll give you a clue. You require an enzyme to convert one form of hormone into another form of hormone where activation happens, and that enzyme is having isoenzymes 1, 2 and 3, and that isoenzyme that is present here in liver is capable of doing this transformation. Let me tell you another clue. The enzyme is deiodinase. Now tell me which hormone is it? Now tell me which hormone is it? It is thyroid hormone. So look at this. We have type 1 deiodinase present in the liver, kidney. And deiodinase can actually deionate both the rings. It tries to convert the T4 into T3. So that when you remove one iodine, it becomes T3. Type 2 deiodinase is found in heart, skeletal muscle, CNS, fat, thyroid and pituitary gland. Type 3 is found in fetal tissue and placenta. But all of them can actually be helpful for conversion of T4 to T3, while type 3 deiodinase activity can only deionate in the inner ring of thyroxine or tyrosine and is a major inactivating enzyme. So here, oh I'm so sorry, I marked 2, type 1, the enzyme that acts on the liver. Next question. Triple H syndrome is due to deficiency of which enzyme? Triple H syndrome is due to deficiency of which enzyme? Take a guess. Arginase, arginosuccinase, ornithine transcarbamylase. Which one? C, B, A. Okay. Now, triple H syndrome is not because of any enzyme deficiency. Let me tell you how. We start with ammonium and carbon dioxide forming carbamyl phosphate. This carbamyl phosphate will get ornithine. All these things are happening in the mitochondria and it can extend into the cytoplasm. When the ornithine reacts with carbamyl phosphate, it produces citrulline. This citrulline comes outside, which reacts with arginine. It reacts with aspartate to form argininosuccinate. That arginosuccinate will break into arginine and fumarate. Fumarate enters into TCA cycle. That is why TCA cycle and urea cycle are combined together. You call them as Krebs-Henselt bicycle. That arginine is broken down by the help of the enzyme called as argininase, which is a hydrolase that will be producing ornithine by releasing something called as urea. Now watch this area. there are certain transporters which will push citrulline out and allow ornithine inside. Now when this see, Padmaja has asked a question. Deiodinase activity can be found in 1, 2 and 3 areas. If at all the liver is failing, the other sources of the deiodinase can actually help in the conversion of thyroxine to, the question was about in the liver, which of the hormones are getting activated. So the answer is staying state that the thyroxine is getting activated in the liver. But thyroxine is not only getting activated in the liver, it can activate anywhere else, even in the heart. So on that process, even though in liver failure, the production of thyroxine activation can be lesser, still at minimal levels, it can keep on happening. In this part, look at this. There is a transporter called as ornithine-citrulline exchanger. Citrulline goes out, ornithine comes in. If this exporter is not functioning, then citrulline stays inside the mitochondria. That citrulline will react with lysine to form a compound called as homocitrulline. Now, the whole reactions are going backwards. Ammonia is getting elevated. You have hyperammonemia. And this ornithine is actually not going inside. It is staying outside because it is not able to enter through this particular translocase. So when the ornithine increases in the cytoplasm, then it comes into the blood. You call it as hyperornithinemia. So I repeat, the triple H syndrome is hyperornithinemia, hyperammonemia. It is not hypercitrullinemia. It is citrulline reacting with lysine to form homocitrulline. So it is called as homocitrullinemia. This is called as triple H syndrome. It is not a deficiency of any enzyme. It is a defect in the transporter. Okay. Darbepoetin is most useful in the treatment of anemia caused by Darbepoetin is most useful in the anemia caused by excellent. It is chronic renal failure. Remember, kidney is the most important source of, yes, erythropoietin. Erythropoietin is important for erythropoiesis, and that is capable of causing anemia of chronic disease. Anemia of chronic what disease? Chronic kidney failure is also a disease of chronic, because of which you have anemia. So darbepoetin is an analog of erythropoietin which can be helpful in the treatment of anemia caused by chronic renal failure. Is that clear? Next question. Tetracycline inhibits protein synthesis by acting in which part of the molecular biology? Let me tell you the answer. T for T. Tetracycline inhibits the 30S subunit and inhibits the binding of aminoacyl tRNA. So this is how the picture looks. Tetracycline will come and bind to the ribosome 30. This is 30S ribosome, this is 50S ribosome. 50S ribosome looks bigger, 30S ribosome looks smaller. And as the tetracycline comes and binds to 30S ribosome, you will not be able to go for further transcription. So tetracycline blocks the ribosomal protein synthesis of bacteria, decrease cytochrome C synthesis, decrease oxidative phosphorylation, increase aerobic glycolysis and lactic acid formation. Is that clear? Increase anaerobic glycolysis and lactic acid formation. The organism dies by lactic acid formation. Let me repeat once again. Tetracycline blocks the transcription and translation by inhibiting the 30S ribosome. Because of the cytochrome C synthesis does not happen, decrease oxidative phosphorylation, lactic acid formation. Which of the following is true about different structures of protein? Which of the following is true about different structures of protein? Okay, let me give the answer. Secondary structure is not a three-dimensional structure. Primary structure is not stabilized by disulfide bonds, it is by peptide bonds. Primary, secondary, tertiary structures are destroyed by denaturation is wrong, because primary is always intact, secondary can be destroyed, tertiary can be destroyed. So the only right answer is super secondary structures are compact three-dimensional protein structure of several adjacent elements of secondary structure. Several adjacent elements of secondary structure means alpha turn beta, alpha helix beta, etc. So this is also a secondary structure, this is also a secondary structure. When multiple secondary structures are alternately being placed adjacently, that is called a super secondary structure. Next, which enzyme is irreversibly inhibited by aspirin? Which enzyme is irreversibly inhibited by aspirin? Generally, your answer will be cyclooxygenase called as COX, but the cyclooxygenase has another name called as prostaglandin H synthase. Remember this. The first prostaglandin to be produced will be prostaglandin H. From there you will be able to derive G. From there you can produce D, E, F, I, etc. Okay. Let's go for the next question. Test to diagnose adequacy of vitamin B1 level is. B1 is thiamine. The test you have to perform to understand the B1 activity is, first you have to follow the thiamine acting as a thiamine pyrophosphate as a coenzyme for which enzyme? The enzyme for which thiamine acts as a coenzyme is transketolase. So you will be doing RBC transketolase estimation. If RBC transketolase estimation is good, then you understand B1 levels are adequate. Now if the transketolase levels are lesser, when you give B1, in spite of that the activity is lesser, then B1 is not deficient, transketolase enzyme is deficient. When you give B1, because of which transketolase activity is increased, then B1 was deficient. By giving B1, you are able to have a better activity. Now on the same line, if you look at this, test to diagnose adequacy of vitamin C level. Vitamin C is seen as ascorbate, and that ascorbate is abundantly found in leukocyte. So leukocyte ascorbate test is a test for identifying the adequacy of vitamin C levels. What about FIGLU test? FIGLU is form-aminoglutamic acid excretion, which is called as histidine load test. Histidine load test. When folic acid is not available, then histidine breakdown actually is deficient, because of which FIGLU excretion happens. Methylmalonic acid in urine is because of B12 deficiency. Obermeyer test is done in case of tryptophan adequacy. That is in Hartnup disease. Let me repeat once again. FIGLU is for folic acid and histidine load test. Methylmalonic acid is for B12. Obermeyer is for tryptophan. Leukocyte ascorbate test is for the vitamin C. Let's go for the next question. The enzymes involved in substrate level phosphorylation are pyruvate kinase, succinate thiokinase, and phosphoglycerate kinase. Phosphofructokinase is only converting fructose 6-phosphate into fructose 1,6-bisphosphate. Here you are utilizing an ATP, but in substrate level phosphorylation you are generating ATP. Generating ATP without entering into electron transport chain. Without entering electron transport chain, if you can generate ATP, that is called as substrate level phosphorylation. The answer is 1, 3 and 4, which is option D. Lipid soluble plasma membrane associated antioxidant. Lipid soluble means it can bind to the plasma membrane and can cross the membranes. The one who can actually act as antioxidant by being lipid soluble is vitamin E. Vitamin E's chemical name is alpha tocopherol. Then if they ask you water soluble extracellular antioxidant, then the answer would be vitamin C. Okay. Next, look at this question here. 3-year-old boy, first degree consanguineous couple, darkening of urine, left to standing. The moment you see darkening of urine, you understand it is alkaptonuria. In alkaptonuria, the enzyme deficiency is homogenesic acid oxidase. Okay. Next question. Estimation of serum bilirubin is done using what? You can actually estimate the serum bilirubin as direct bilirubin or indirect bilirubin or total bilirubin by performing Vandenberg reaction. Vandenberg reaction, direct positive means it is directly reacting with it. It means it's a conjugated bilirubin. If I go for indirect positive, it means what? When you throw the bilirubin inside, it won't directly give you an answer. So you have to add methanol. After adding methanol, if the test is coming out positive, then the bilirubin is unconjugated bilirubin. So conjugated bilirubin is direct bilirubin and unconjugated bilirubin is indirect bilirubin. Both can be done by Vandenberg reaction. What is the function of Fauchet's reaction? Fauchet's reaction can show the presence of bilirubin and biliverdin. It is only showing the presence or absence, qualitative test. While Ehrlich's test is done for the presence of urobilinogen and Schleisinger's test is done for the urobilin. I repeat, Ehrlich's test is for urobilinogen while Schleisinger's test is for urobilin. Okay. Next, Leach-Nyhan syndrome is caused by deficiency of, it is HGPRTase deficiency. If there is a complete deficiency of HGPRTase, then there is complete deficiency of a salvage pathway. What is salvage pathway? Whenever you have the GMP or IMP or AMP after reaching its normal activity, when their expiry date has come, they should be broken down. If you allow them to break down, if they completely come to form uric acid, then to synthesize one more adenosine monophosphate, it takes an extremely difficult pathway. You have to start from the scratch to produce AMP. Instead, if you allow the AMP or IMP or guanine to go back to this guanine and hypoxanthine or adenine, and then with the help of PRPP and HGPRT, if you can retain this GMP, IMP and AMP, that is called as salvage pathway. For example, if you actually have generated 100 rupees buck activity, if you break them down to 1, you don't allow them, you break it only till 70 percentile and then take it back to 100, you will lose only less amount of energy to regenerate the whole activity. The 100 percentile activity can be regenerated at the level of the 70 percentile itself. You don't have to allow it to break down till the end. So that is called as salvage pathway. If that salvage pathway is deficient, you completely deficient, it's called as Leach-Nyhan syndrome. If there is a partial deficiency of HGPRTase enzyme, that is called as Kelly-Siegmiller syndrome. Okay. Yes. Next question. If a sample of DNA, in a sample of DNA, adenine is 28%, what will be the amount of cytosine? So, imagine the total is 100, out of which adenine takes 28. Adenine always binds and double bond with that of thymine. It means thymine also should be 28. So, it means 28 plus 28 automatically becomes 56. They are asking you for cytosine. It means cytosine will make a triple bond with that of guanosine. So, cytosine and guanosine should be equally the same. Now, if adenine and thymine together have taken up 56%, then remaining is 44%. And that 44% is equally shared by both cytosine and guanine, which is 22% each. The answer is 22%. Identify the vitamin which on heavy doses can cause cystoid macular edema. The one vitamin which is given in case of dyslipidemia. In patients suffering from dyslipidemia, the vitamin that is given would be vitamin B3 niacin in the form of nicotinic acid. The nicotinic acid by studies has proven very well. HDL levels has been elevated and LDL levels have been decreased by taking nicotinic acid. When nicotinic acid is taken in huge amounts, then it can cause ocular manifestations like cystoid macular edema. Other problems it can cause would be toxic amblyopia, proptosis, eyelid edema, blurred vision, and superficial punctate keratitis. Of all of them, the most dangerous is cystoid macular edema. True statement regarding Huntington's chorea is, in Huntington's chorea, there is increased number of CAG repeats, which is technically trinucleotide repeat expansion type of disorder. So this is the true statement here. Just give me a moment. In Huntington's chorea, you will be having a CAG repeat, because of which you will be having a gain of function mutation. It's an autosomal dominant kind of disease. And so you will be having mood disturbances, severe dementia, involuntary jerky movements. A more concentrated source of cobalamin. Cobalamin's better source will be non-vegetarian food, which is technically meat which contains liver component, and also it can be a source from the bacteria present in your gut. The only B complex vitamin which can be stored in the liver will be B complex, that is B12. Okay, next, these are remaining high quantities: beef, liver, chicken, fortified breakfast cereal, low fat milk, yogurt, cheese, eggs, fish, shellfish, trout, salmon, tuna fish and clams. Okay, blood stored in citrate phosphate dextrose is better for road traffic accident patients compared to acid citrate dextrose. Why? In case of citrate phosphate dextrose, the 2,3-BPG fall is lesser. It means 2,3-BPG is slightly higher. Because of which, when you go for the oxygen dissociation curve, it will be towards the right side. Whenever the oxygen dissociation curve is towards the left side, it means the hemoglobin is holding the oxygen tightly. If the oxygen dissociation curve is towards the right side, then hemoglobin is releasing the oxygen. In road traffic accident patient, stored blood is not supposed to be used in general. Fresh blood should be given. Why? Because if it is stored, then the oxygen dissociation curve should be going to the left. In case of acid citrate dextrose, the oxygen dissociation curve is leftward movement. In case of citrate phosphate dextrose, the leftward movement is slightly lesser, is slightly to the right side. So it can release the oxygen to the tissues. That is why it can be more helpful. The drug that is known to inhibit DNA replication is 6-mercaptopurine. 6-mercaptopurine can help in the DNA replication blockage. They can act as antimetabolites. The other antimetabolites are folate antagonist called as methotrexate, purine antagonist called as mercaptopurine, pyrimidine antagonist called as 5-fluorouracil. Werner syndrome, which can lead to premature aging, can cause progerias. Those progerias are caused due to a defect in the DNA helicase enzyme. This is how it looks. A lady, this is from a Japanese picture. This is a lady who was very young at 18 years of age. By 40 years of age, she becomes looking older. The 19 years or 18 years old girl who looks even more older, faster. Progeria happens. Identify the feature which is not a part of metabolic syndrome as given by NCEP 83 guidelines. According to the NCEP 83 guidelines, you will not be having high LDL as a mark. Hypertension is a part, hypertriglyceridemia is a part, central obesity is a part. What else are seen? Abdominal obesity, waist circumference given for males and females, triglycerides more than 150, HDL cholesterol for men less than 40, women less than 50, blood pressure more than 130 by 85, fasting glucose more than 120, 110 are all markers for metabolic syndrome. Hepzidin's role lies in what? Hepzidin, Sidel, is equal to killing. Hepzidin technically decreases the transcription and translation of ferroportin. Ferroportin helps in transport of iron into the body for absorption. So when hepzidin is present, it decreases iron absorption. Okay, transfer of iron to hepatocytes is the place where it has a negative role. Hepzidin. Proteoglycan present in glomerular basement membrane. In the glomerular basement membrane, there are 4 components. One is laminin, collagen, heparan sulfate, nitrogen. Among the 4, the heparan sulfate is the only proteoglycan. And the glomerular membrane can have a capillary endothelium, basement membrane, and Bowman's visceral epithelium, which is called as podocytes, out of which the heparan sulfate is the one that is capable of acting as the proteoglycan. And it can offer the negative charges. And you will be having glomerular basement membrane polyanions. If those polyanions are lost, that is called as nephrotic syndrome. That is what is called as nephrotic syndrome, where there is minimal change disease because the doors are open, proteins are lost massively, which is called as nephrotic range massive proteinuria. Nephrotic range massive proteinuria. Okay. The clotting factor to decrease gamma carbogylation of glutamic acid residues would be following warfarin therapy. When you give warfarin, it is capable of inhibiting the vitamin K epoxide reductase, because of which vitamin K cycle is inhibited. When vitamin K cycle is inhibited, then vitamin K dependent gamma carboxylating proteins are inhibited, which is 2, 7, 9, 10 proteins like C, S, Z, out of which you will realize the one that will be maximally suffering would be factor 2. Identify the apoprotein proven to be associated with increased risk of Alzheimer's disease. If you look at it, apoprotein E, you have the apoprotein E2 that causes decreased Alzheimer's disease risk. Apoprotein E2 causes decrease in the Alzheimer's disease risk, while apoprotein E4 will increase the Alzheimer's disease risk. These are the information you should carry for your exams. Apoprotein E2 will decrease the risk of Alzheimer's disease. Apoprotein E4 will increase the risk of Alzheimer's disease. Which of the statements regarding mitochondrial DNA is false? Mitochondrial DNA is double-stranded, inherited from mother. All respiratory proteins are synthesized within the mitochondria itself. High mutation rate. It is double-stranded, yes. It is circular, yes. It is completely from the mother, yes. That is why the ovum is the one who gives the mitochondria. The mother is the one who is actually giving you. All respiratory proteins are synthesized in the mitochondria? No. Mitochondria can have its own DNA from which most of the respiratory enzymes, that is electron transport chain enzymes, can come. But there are some other electron transport chain proteins like cytochromes who will come from the nuclear DNA. That is why option C is wrong. It has a high mutation rate because in nuclear DNA, when you go for DNA replication, you have the polymerases who have proofreading mechanism. That proofreading is very less and minimal in case of mitochondrial DNA, so mutation rate is very high. Which receptors are blocked immunologically in myasthenia gravis? In myasthenia gravis, autoantibodies will be formed, which will be an equivalent of type 2 hypersensitive reaction, where they come and block the acetylcholine activity. When acetylcholine receptors are blocked, there is decreased excitation. If there is decreased excitation, there is decreased contraction. The patient can go for flaccid paralysis. Tyrosinosis is caused due to deficiency of which enzyme? When I say tyrosinosis, it can be of two types. Tyrosinosis type 1 and tyrosinosis type 2. If it is type 2, then the enzyme involved is tyrosinase or tyrosine hydroxylase. That is T for T, 2 for T. And type 1, 1 is first. First is fumaryl acetoacetate hydrolase. This is the enzyme deficient for tyrosinosis. Which of the following regulates the cerebral blood flow? Which of the following regulates the cerebral blood flow? Cerebral blood flow is best regulated by PO2, arterial PCO2, potassium ions, adenosine. All four can regulate the cerebral blood flow. But the BP is not a very strong regulator of the cerebral blood flow. Generally, the BP between 60 and 150 millimeters of mercury will not actually cause much of a difference. Pick the correct combination of statements for a competitive antagonist. Competitive antagonist, I just now told you the reason. We have already discussed this question, where what remains constant? The Vmax remains the same, but the Km value increases. The intestines specifically express apoprotein B48 instead of apoprotein B100. This is due to what? In the intestines, you will be having RNA editing. That is done by an enzyme called as cytosine deaminase or cytidine deaminase, where in case of CAA, this cytidine undergoes deamination. Suddenly UAA will happen, while the whole reading of the whole stream, the reading of the strand happens at the 48th percentage. When the whole polymerase is reading at the percentage where the transcription is happening, suddenly when the translation is supposed to happen, instead of reading CAA, they start reading UAA. UAA is a stop codon, so at the 48 percentage it stops, and remaining protein is called as B48 because it is only 48% of the B100. This is happening by RNA editing using the enzyme cytidine deaminase, which converts CAA into UAA. Look at this question here. A middle-aged male presents with easy fatigability, exertional dyspnea, acute weight loss, frequent falls, and the hemoglobin is lesser, bilaterally diminished vibration sense. Local physician administers folate, and his blood picture anemia has improved. Anemia improved, but vibration sense did not improve. Why? Remember, both B12 deficiency and folate deficiency both can cause the same picture called as megaloblastic anemia, and that is the peripheral picture. But the B12 deficiency, in addition to causing megaloblastic anemia, can cause severe combined degeneration of your spinal cord and also can cause neurological manifestation. When you give folate, the megaloblastic anemia can be corrected. But because you think the megaloblastic anemia is corrected and you stop giving the peak treatment, then the B12 deficiency can lead to severity of the neuronal degeneration. So folate therapy causes rapid use of B12 stores, aggravating symptoms, because of which the CNS disorder is not stopped, but the only peripheral smear is stopped. So give both folate and B12 together. The term polyuria is used generally when the everyday urine output is how much? When the everyday urine output crosses 50 ml per kg per day, you call it as polyuria. What is the minimum amount of urine to be produced every single day? Every single day, 500 ml per day should be produced as urine for normal healthy life. Minimum 500 ml. Among the following chemicals, which one is used in preserving or storing the viscera in the toxicological analysis? Rectified spirit can destroy the proteinaceous component of viscera. Glycerin is not useful. Formaldehyde and formalin can actually cause destruction or erasure of all the, erasing of the evidences. So saturated salt solution is the best chemical for storing the viscera for toxicological analysis. 6-year-old boy keeps suffering from recurrent chronic infections caused by capsular bacteria. Of the following, which one is deficient? Remember, encapsulated bacteria are best handled by the opsonization procedure where spleen will come into picture. Along with that, IgG2 is the one who does the best activity. After complex formation, IgG2s can trigger complement independent macrophage and PM and mediated phagocytic activity, particularly against the encapsulated bacteria. Among the following, which of the following acids ingestion is not a contraindication for gastric lavage? Remember, HCl, sulfuric acid, nitric acid, all the three can be called as vitriolage if it is attacked for the self or the others. Carbolic acid is only, what is carbolic acid, people? Can anybody tell me what is carbolic acid? Excellent. Dilute phenol is called as carbolic acid. This will not be a contraindication for gastric lavage. A 40-year-old female has a thyroid swelling. On examination, TSH levels are found to be elevated. Post-operative cytology showed lymphocytic infiltration with Hurthlase cells. What may be the condition? We'll finish the class in just 10 minutes. In 10 minutes, we'll finish the class. Okay? In 10 minutes, we'll finish the class. The TSH levels are elevated. What does it mean? If TSH levels are elevated, you can consider them as hypothyroid state. Of the given conditions, Hashimoto's thyroiditis is the only hypothyroid condition, and Hurthle cells are very commonly found in case of Hashimoto's thyroiditis. They're also found in other kinds of carcinoma. But in case of carcinoma of thyroid, you may be having hyperthyroidic activity, but Hashimoto's thyroiditis can show you hypothyroidism. 30-year-old female complains of fatigue and tenderness. The hemoglobin values are 8 gram percentage, MCV of 101 femtoliter. Peripheral examination shows microcytic erythrocytes and ribosomal precipitates are seen inside the basophils. The ribosomal precipitates found inside the basophils are nothing but basophilic stippling. The basophilic stippling is because of the precipitation of the RNA proteins inside the basophil. Why? Whenever you have a protein and that protein has a disulfide linkage, one of the methods of denaturing a protein is by adding a heavy metal. A heavy metal can actually destroy the disulfide linkage. For example, if I introduce lead, if a protein is supposed to have a disulfide linkage, the disulfide linkage will break and lead sulfur complex will form, because of which the protein will lose its activity. When the protein loses its activity, then that is referred to as precipitation or denaturation. So this is what is referred to as lead poisoning. From now onwards, when somebody asks you what is basophilic stippling, you please tell them addition of heavy metals can cause disulfide linkage breaking. Disulfide linkage is the covalent linkage found in case of tertiary structure of a protein. Primary structure covalent linkage is peptide bond. Tertiary structure covalent linkage is your disulfide bonds. When the disulfide bonds are protein, the precipitation happens, denaturation happens. That precipitation is called as basophilic stippling. The drugs imatinib and sunitinib are known to act as anti-cancer agents by what biochemical or molecular changes? Do they act as inhibitors of tyrosine kinase, protein kinase A, IP3DAG pathway, or ADP ribosylation of EF2? Which one? You all know the answer is tyrosine kinase, where cross-phosphorylation of the receptors can happen, and ADP ribosylation of EF2 inhibition is the property of diphtheria toxin. Diphtheria toxin. In which of the following abnormalities does the serum calcium levels get elevated? Try this answer. Try this question, please. In which of the following abnormalities does the serum calcium level gets elevated? We can speak about multiple complex ways by which you can arrive at the answer, but the simple thing is when a parathyroid gland has gone for a tumor, because of which the parathormone has increased. The parathormone increases the calcium massively. So if you want to bring the calcium down, you have to again elevate the calcitonin production. So serum calcium elevation can happen because of hyperparathyroidism in the simplest ways possible, even though they not be technically very clearly straightforward accurate, but this is the whole idea about the logic. So that can happen in case of medullary thyroid cancer where the parathormones can be elevated. Filgrastim is a drug used to treat which of the following conditions? Is it viral infection, anemia, neutropenia, malaria? Which one? Excellent. It is important in case of neutropenia, especially in case of febrile neutropenia. These filgrastim, monograstim are all drugs which are useful as granulocyte colony stimulating factors. They are the ones who act as granulocyte colony stimulating factors, and they will increase the production of neutrophils. C-peptide seen in which condition? In subcutaneously injected insulin, in proinsulin, as a combined entity with insulin after secretion, a gastrointestinal proactive molecule. Which one? No, not as a combined entity with insulin secretion after secretion. It's not after secretion. C-peptide is usually found in the proinsulin. C-peptide is found in the proinsulin. Look at this. This is the pre-proinsulin. B chain is here. A chain is here. Between the B chain and A chain, you have a C-peptide, and then you have an N-terminal signal peptide. Now, when you go for endoplasmic reticulum of beta cells, you have a disulfide linkage where the particular N-terminal peptide is removed. When the N-terminal peptide is removed, what you have is proinsulin. The proinsulin contains the green colored bigger one, C-peptide. And when they go for the Golgi apparatus of the beta cells, the C-peptide is removed. Then you have insulin, who has a half-life of 6 minutes. As long as they were bound, the half-life was very long. When the C-peptide is removed, the half-life becomes very less of 6 minutes. Okay. The hormone vasopressin acts on which of the following in the collecting duct? The answer is aquaporin 2. The hormone vasopressin acts as, vasopressin is the other phase of one more hormone called as antidiuretic hormone. The vasopressin will act on aquaporin channel 2. And when they act here, that is V2 receptor, they will cause activation of GS alpha, and that will activate the ATP and cyclic AMP, because of which aquaporin 2 will be upregulated. I repeat, when the vasopressin comes and sits on vasopressin 2 receptor, because of which there is ultimate activation of aquaporin 2, then aquaporin 2 will be upregulated on the surface, which is the apical plasma membrane. Drug acting on potassium channels include which of the following? Okay. What is the answer? Drug acting on potassium channel includes which of the following? Yes, the answer is nicorandil. Nicorandil acts as a potassium channel opener and also acts as a nitric oxide releasing property. It has two properties. One is to release nitric oxide. The other one is to act on potassium channels. What kind of potassium channels are they? They are the ATP sensitive potassium channels. And because of the ability of releasing nitric oxide, they can act as an ischemic preconditioning agent and also as a coronary dilator. So they can be helpful in case of angina. Okay. Next, conversion of norepinephrine to epinephrine is mainly by which of the following components? Excellent. Norepinephrine means no R in the epinephrine ring. Epinephrine is an example of a catecholamine. Catechol amine means they contain catechol nucleus. Who is the catechol nucleus? When OH is present on the phenylalanin's benzene ring, then that is referred to as the catechol nucleus, and it begins with tyrosine. So when there is no R, there is no methyl group in the epinephrine. If you add the methyl group, that becomes epinephrine, and that methyl group is offered by S-adenosine methionine. When the S-adenosine methionine comes near the norepinephrine, norepinephrine becomes epinephrine, while S-adenosine methionine becomes S-adenosine homocysteine. This is done by phenylethanolamine N-methyl transferase enzyme. It is an example of a N-methyl transferase enzyme. Why? Because you are transferring the methyl group to the N-nitrogen. Okay. In lipoprotein lipase deficiency, which of the following is increased following a heavy meal rich in lipids? If you take a heavy meal which contains a lot of lipids, the lipoprotein lipase is capable of breaking it down. So the meal means exogenous lipid. Exogenous lipid contains exogenous triglycerides, and they are placed inside the chylomicron. If there is a lipoprotein lipase deficiency, then chylomicrons will be elevated. Why? I repeat once again, from the food that you eat, the exogenous triglycerides will be attempted inside the chylomicron. The chylomicron should be broken down by the lipoprotein lipase at regular intervals. If this enzyme is not there, chylomicron is not broken, so it can produce type 1 hyperlipoproteinemia, also referred to as hyperchylomicronemia. Hyperchylomicronemia. Okay. Next, Zellweger syndrome. It is not Zellweger syndrome. It is Zellweger syndrome. Zellweger syndrome is also referred to as infantile Refsum's disease. What is the point? You have Refsum's disease separately. You have infantile Refsum disease separately. Both of them are involved in very long chain fatty acid oxidation. Understandable. But in Refsum disease, inside the peroxisomes, you don't have the enzyme called as phytanic acid oxidase, because of which phytanic acid accumulates, which can be dangerous for the brain. Mental retardation happens. But in infantile Refsum disease, the enzyme deficiency is not the problem. Either peroxisomes are absent, or the fatty acids are not targeted. There is a targeting defect. They are not targeted to the peroxisomes. That is, the enzymes are already present. The enzymes are not traveling into the peroxisomes, and the fatty acids are not traveling to the peroxisomes. This is called as infantile Refsum disease, which is a Zellweger syndrome. It's a type of a defect in the peroxisomes. Okay. Identify the protein which is not synthesized in the liver. Liver is the site of synthesis of albumin. Liver is also the site of synthesis of protein C, protein S, and protein Z involved in the coagulation. Liver is also the source of acute phase reactants when there is inflammation. For example, when somebody has inflammation, even during the COVID period, you had the interleukin 6 estimation, you had the ferritin estimation. Ferritin acts as a acute phase reactant, and also when you look at CRP levels, all of them are acute phase reactants. They come from the liver. But immunoglobulins will come from the B lymphocytes at low concentrations, and when the same B lymphocytes become plasma cells, they come from the plasma cells at high concentration, immunoglobulins. Okay. Which of the following amino acid is involved commonly in the formation of creatinine, nitric oxide, and also urea? It is not creatinine, it is creatine. Let me make it very faster. Glycine plus arginine will form guanidinoacetic acid. This guanidinoacetic acid will come across S-adenosine methionine, which becomes S-adenosine homocysteine. On methylation, methylated version of guanidinoacetic acid is referred to as creatine. Also remember, nitric oxide is coming from that of arginine. Arginine, on breakdown by arginase, can produce nitric oxide. Urea is coming from arginine when the arginine breaks into ornithine and urea in the urea cycle. So the common amino acid is arginine. We are coming very close. We have only five more questions. Please be with me. Which of the following enzyme is the odd man out with respect to the reducing equivalent generated? Here you have isocitrate dehydrogenase, malic enzyme, G6PD, ATP citrate lyase. We are focusing on the reducing equivalent, right? Reducing equivalents can be either NAD plus or NADP plus or FAD plus. Here you have malic enzyme G6PD and isocitrate dehydrogenase if it is cytosolic. All the three are known to be synthesizing agents for NADPH. I repeat, isocitrate dehydrogenase cytoplasmic, cytoplasmic isocitrate dehydrogenase, malic enzyme, G6PD, glucose 6-phosphate dehydrogenase. All three enzymes are involved in the synthesis of NADPH, while ATP citrate lyase is not actually involved in the synthesis of NADPH. Is that clear? Either type 1 or type 2 hyperammonemia can slow down the TCA cycle by removing or reducing which are the following intermediates. Whenever ammonia levels are elevating in the brain, then your alpha ketoglutarate will try to trap the ammonia to form glutamate. In the brain, it goes one step further to accept one more ammonia to become glutamine. So whenever there is hyperammonemia, for every ammonia molecule elevated, alpha ketoglutarate will be utilized. So when the alpha ketoglutarate is disappearing, then one TCA cycle disappears. Because of decreased TCA cycle activity, because of disappearance of the alpha ketoglutarate, the brain will not have enough amount of, the brain will have sudden deficiency of ATP. Why? Because one TCA cycle is worth 10 ATP. So sudden fall in ATP can lead to encephalopathy. One more reason, whenever alpha ketoglutarate becomes glutamate, glutamate acts as an excitatory neurotransmitter, because of which you can expect seizures. That is also a part of encephalopathy. Also remember, in case of hyperammonemia, ammonia is a very hygroscopic compound, attracts water. Because of water attraction, edema can happen. That edema settles by gravity at the base of your brain, because of which brainstem compression can happen. These are all the features that can happen when a patient suffers from hyperammonemia. Okay. Next, which of the following enzyme is deficient or with reduced activity in case of Menkes disease? In Menkes kinky hair syndrome, lysyl oxidase is the defective one, and this is actually ATP gene. It is ATP acid binding protein, which is in case of Wilson's disease, it is 7B2. Here it will be in case of Menkes disease, it is 1. Okay, remember this is 1. Lysyl oxidase. Deficiency of the enzyme LCAT can cause increase in which are the following? LCAT is lecithin cholesterol acyl transferase, and lecithin is a phospholipid. Phospholipid is abundantly present in the HDL. So lecithin cholesterol acyl transferase will be able to transfer acyl groups from here to elsewhere. If it can actually be deficient, then HDL values will be increasing. HDL value will be increasing because from HDL you will not be able to remove the fatty acids. Now look at this question. I think this is almost the last question. Two more questions. A 45-year-old alcoholic presents to the OPD. 45-year-old alcoholic presents to OPD with edema, visual disturbance. Mental state change is seen. BP is elevated. He has high output cardiac failure. Which of the following vitamins is given in the picture? This is vitamin B1 deficiency, seen as beriberi. And what beriberi it is? It is Soshin beriberi. Why is it Soshin beriberi? Because it is a wet beriberi. Why is it wet beriberi? Because you understand this is a case of cardiovascular beriberi where there is a high output cardiac failure. And this edema and visual disturbance are all properties of the B1 deficiency. It happens generally in alcoholic people. So you will be having a concept called as GOA MP. GOA is global aphasia can be noticed. O will be ophthalmoplegia, because of which visual disturbance can be noticed. A is ataxia. M is mental disturbances. And P is peripheral neuropathy. If the B1 deficiency becomes worse, then it can lead to Wernicke's encephalopathy and Korsakoff's psychosis. Together they are called as Wernicke-Korsakoff syndrome. And here remember, This alcoholism is the reason for B1 deficiency. Why? Because in alcoholism, the calories will be high, nutrition will be low. When the calorie is high, the patient will not be hungry. Because he is not hungry, he won't be able to eat proper nutritious food. And even if he eats nutritious food, the absorption is very poor in alcoholic patients. That is why B12 deficiency. Next, elevation of glutamine in the CSF, blood and urine is most probably due to the defect in which of the enzyme? The answer is CPS1. When the CPS1 happens, deficiency happens, then you will be having carbon dioxide plus ammonia being elevated. And when the ammonia is elevated, the alpha ketoglutarate will become glutamate, because of which glutamine increases. So glutamine increases in CSF, blood and urine. Why CPS1? Because when carbon dioxide plus ammonia itself are not actually getting trapped, then free ammonia is very, very high. That is called as hyperammonemia type 1. Next one. The type of cholesterol that accumulates as gallstones is crystalline cholesterol monohydrate, not even dihydrate. Look at this part. You will be having gallstones composed of undissolved air. So cholesterol monohydrate crystals are seen. They are crystalline in nature. Okay. So yes, glutamine only in brain. That is why I am saying they are accumulating in the CSF first, then it will flow off into the blood and ultimately come out in the urine. The activity happens in the brain. When the glutamine enters into the brain CSF, from there it can come back into the blood and enter into the urine. The source of synthesis of glutamine is in the brain, but the glutamine overflows into the other parts of your body. Okay, so with this, we conclude this 100 MCQs marathon, and I have done all the 100 MCQs as promised. So I hope all of you will be able to use this information in your exams. At least out of these 100 MCQs, even if 2 MCQs or equivalent can actually come out in addition to what you know in your exams, your ranks will jump by at least 10,000 to 2,000 if you're in the rank of 20,000. Your ranks will be jumping very faster if you are in the range of within 1,000. So I hope your ranks are getting better, will get better at the end of the session. And if you like the class, please put a thumbs up. It can help more students watch it. Thank you very much. Good night. All the best for your exams. And when I come back with the 100 MCQs booster class for microbiology, I'll let you know on the groups. Good night.